technology. I welcome you once again, all of you, to this last day of our international conference come workshop. I hope the 2 days were fruitful for every one of you and it was enriching to every one of you. Now, for the third day, we have Dr. Vinay Kumar as our speaker for the talk. Let me introduce Dr. Vinay Kumar. He is presently Director, Translational and Developmental Science at Coherus Biosciences, San Francisco, USA. He is responsible for the safety assessment of various novel therapeutic compounds and MABs currently in development for the immuno-oncology program. He has earned a PhD degree in biochemistry from Charles University, Prague, Czechoslovakia. His doctoral thesis was focused on post-translational modification in soluble recombinant therapeutic proteins secreted by lower eukaryotes. After finishing his doctoral thesis, he worked at Johns Hopkins University, Baltimore, and Tulane University, New Orleans, Los Angeles on development of transmission blocking vaccines against malaria and molecular pathology of HIV or SIV enteropathy as a postdoctoral fellow. He also studied the impact of cannabinoid, epigenomic, and miRNA mechanisms on HIV disease progression using rhesus macaque model. His research interests are cancer immunotherapy, vaccine development, and molecular pathology of HIV. I welcome you, sir, to this conference and would like to invite you to deliver your talk. Over to you, Dr. Vinay. You are muted.
Oh, can you hear me now?
Yeah. Yes.
Sorry.
Yes.
Sorry for the trouble. So thank you very much for the invitation. First of all, I would like to thank the organizers, especially Dr. Tabassum Jehan and Dr. Ruchi, who gave me this invitation. And Dr. Ruchi has been in contact with me for regarding, various aspects of this presentation. So thank you so much. I'm very excited to be here and to talk about a little bit today, as you said, that I work in cancer immunotherapy and how we can use certain in silico tools, how they can help us in narrowing down the window and save us quite a lot of money in the long run. And I will go through that in my presentation. So I'm sharing my slides, and please let me know when you can see them.
Thank you so much, Dr. Vinay. I would like to take few questions. If any participant has question, they can ask, they can raise hands, or they can write in the chat box. Meanwhile, I have few questions with me that have come. Yeah. One question is, what is the percentage of effectiveness with in silico approach?
It depends, case to case, it's case on case basis, and most of the time they are pretty accurate because the algorithm has evolved regarding the T effector and T regulatory, the regitopes. There is always a chance that it will there were some concerns that it generally overestimate, but I think it's much better to know that it's a high risk. You can work around on different, work around different options to de-immunize it, than basically going blind-handed like Pfizer did, and they have to shut down the development of that particular program. So they are highly effective, and the good thing is FDA accepts them. Once they provide you the report, regulatory agency take that in account, and if it is shown that it's a low risk, they accept it.
Thank you, Dr. Vinay. Another question, what is the minimum time and phases for clinical trials that are needed to release a biotherapeutic compound, especially in oncology?
You have to have a good phase 3 data at least. So it start in discovery. I will just kind of summarize the whole process. It start in discovery. You start in the research lab as we do in universities. So research department, they come up with a list of candidates against a particular, target. Then from there, from discovery, go to preclinical. Once they have a good idea of, like, let's say they start with 20 and they come with 4, then all these 4 will go in either monkeys and mice. FDA asks you to do at least 2 relevant animal species. Once you have that preclinical data, the animal data, then you, depending on that, you start with phase 1, phase 2. These go together, and finally phase 3 studies. Once you have good phase 3 data, then basically you can get the approval, yay or nay. Once you have a good preclinical data, you go to FDA, you start the new drug application, and after that you are in continuous kind of communication with FDA, showing them the results, what you are doing. So phase 3, at least phase 3, yeah, good phase 3.
Okay. There's a question from the students that how can one get training in this field?
So as I told you, there are some free tools available. If you see immunogenicity prediction on Google, you may find. However, the one we use, it's proprietary from Epivax. It is kind of on expensive side. For example, if you have a couple of molecules, they can do it for you, and they charge you like $50,000 to $60,000 per molecule. They also license this tool to the big pharma, and they charge like $100,000 per year as a license fee. So this is kind of expensive business, but there are some online tools available you can use. They're not as effective, but they're pretty good.
Okay. One more question is there. What is your take on, as you said, that at least few phases of clinical trials they are required and minimum time is required for releasing of any. So what's your take on the vaccines that we have got in market for the corona that were released in terms of the clinical trials?
Well, that's a very relevant, very contemporary question, I would say. So I think with the mRNA vaccine, so I got personally the Pfizer one myself, so I can talk about those. So it's a new platform basically, and there were concerns about it, but it's not traditional. So there was no animal data involved. It's a mRNA vaccine. It's going to go directly in human. Was it rushed? Yes, it was. Uh, if you have to choose little bit of kind of adverse reaction versus death from corona, so it's always risk benefit analysis in pharma. So I think yes, they were rushed, but I think the data when they released it, the vaccines were released, it was limited. More and more data is coming now since the large populations have been immunized, especially the Israel, the almost whole countries immunized, and we have pretty good data from there. So I think in the end, it was not that bad. But yes, it was high risk. I was concerned myself. But what do you do when you're in the middle of pandemic? You get what you get.
Yes, there's always a trade-off between
Exactly.
So I think that's all the questions that were there with me. If any participant is willing to ask a question, they can just raise their hands. Okay, now I take this opportunity to thank you, Dr. Vinay, for sparing your time. And I know the time lag between India and there in US is pretty much, almost 12 to 13 hours, I guess. So there must be late in the night that you were there for us to, give this interesting and insightful talk. And it was really very interesting and imbibing and informative talk, Dr. Vinay. And I thank you for giving this insight into the whole journey of developing a biotherapeutic tool or a compound and into such an important and, a much needed field of oncology. So thank you so much, Dr. Vinay. And we look forward whenever you are in India, in Delhi, we look forward to have a talk with you offline.
Thank you. Thank you very much, Dr. Jehan. Thank you so much. Thanks, everybody. Have a nice day. Bye-bye.
Thank you, sir. Now, so I would like to take this opportunity to welcome our next speaker for the day, that is Dr. Brijesh Rathi. Welcome, sir.
Thank you. Thank you so much.
Yeah, and we have opportunity of hearing Dr. Rathi earlier also in one of our conferences, and we got overwhelming response from the students and the participants. So now we once again have this opportunity to hear you from you. And I would just like to introduce Dr. Brijesh Rathi before he can deliver his talk. Dr. Brijesh Rathi is obtained PhD degree in chemistry from University of Delhi and currently working as assistant professor of chemistry in Hansraj College, University of Delhi. He was trained as postdoctoral fellow at Massachusetts Institute of Technology, Cambridge, USA. He is recipient of the prestigious Young Scientist Fellowship, Early Career Research Award from DST, Government of India, and Cape Sprint Fellowship from Ministry of Education, Brazil. He also received the prestigious Excellence Award for in-service teachers in 2018 from University of Delhi. Dr. Rathi is an affiliate faculty at University of Loyola Chicago and present and at University of Debrecen, Hungary. He has published more than 90 papers in various journals. He holds the position of section editor for current topics in medicinal chemistry and associate editor membership of chemical biology letters. Dr. Rathi has supervised 1 doctoral and 5 undergraduate research students in the past and currently supervising 5 doctoral students. His main research interests are centered at the development of new antiparasitic and antiviral agents and their target identification aided with the computational techniques. Though the list is quite long, but I would like to now stop here and would like to invite Dr. Rathi to give his talk. Over to you, sir.
Thank you, ma'am. Thank you so much. In the beginning, I would like to thank all the organizers, Zakir Hussain College, for this beautiful, wonderful conference. Of course, certainly this is my second time that I have been giving a talk to you all guys. Maybe students were quite encouraged last time also, and I believe that though I was a little scared, I don't know how I'm going to make a difference this time and maybe more impactful to the students so that it could be very useful. So thank you, and I congratulate the convener of the conference, Professor Tabassum Jahan, and organizing secretary, Dr. Savindra Kumar and Dr. Rahman, and also the coordinators, Dr. Ratnam and Dr. Ruchivir, who is a good friend of mine. So thank you, ma'am, for the opportunity. So today, I take this opportunity to talk a little bit what we have been doing, what we have done in last 5-6 years towards the drug discovery against malaria disease. So here we'll talk about today the how we have utilized the computational techniques for the discovery of multi-stage anti-malarials. Certainly, the term multi-stage anti-malarials is going to be new for all of you, I believe, but it will be very interesting to know about it. Simple chemistry, but high-valued molecules. Ideally, the chemist, because I'm a chemist, they talk about a lot of complex chemistry. So don't be afraid, I'm not going to talk much of very complex chemistry, rather very simple chemistry, because the drug discovery requires very simple chemistry, very simple molecules, but of high weightage, high value, so that they can be easily synthesized and given for the needed as a drug or therapeutics. So certainly the drug discovery is not a piece of cake. It is a very lengthy process. I think all the speakers who are much more, expert than me, they have already given a feedback on it, and they must have intimated every slide and every people that drug discovery is a very difficult task, very lengthy process. So it's something like that. You have to kiss several frogs before you meet the prince. Now, why it is very difficult? Certainly, it involves a lot of steps. Already, Vinay sir has, given his talk, and he was telling about all the trials. So even before trials, you need to have several steps completed. That is, first you identify a disease and target validation, and also you try to find some hit molecules, which you call maybe them very potent molecules, and then you go for lead optimization. Ideally, people try to exit from there and they get good publications. But my point of thinking and my efforts are always beyond that. So we have entered to next level of studies, that is preclinical studies, and also now trying for clinical studies. So that I will discuss how difficult it is, and it's not easy task to enter into the clinical or preclinical studies. So I talk about malaria. Malaria certainly, even now, it is a big problem for many countries across the world, including India, and there are various species known. So I talk about here Plasmodium falciparum, which is a main cause for human malaria. And also Plasmodium vivax, it is also causing human malaria in some of the places, particularly in Brazil. So here, very simple process, what we understand when we talk about malaria is degradation of the hemoglobin. It's not very simple process. It is a very complex, huge process, involves a lot of steps, lot of enzymes, and then it degraded into hemoglobin. So still people have not understood completely, neither biologists, and forget about the chemist. So though we have a lot of therapeutics available so far, that I go to next slide. Yes, anti-malarial therapy. So we know that chloroquine is a very potential therapeutic available to treat simple malaria. Even it is effective in malaria cases to treat even in India. But gradually what has happened, that the chloroquine, including other all which I have listed here, a range of therapeutics, they have gradually started revealing the resistance. And that was a major problem for the anti-malarial therapeutics. Gradually, people have started thinking of novel therapies, and one of the therapy is the frontline therapy, that is artemisinin-based combination therapy. Artemisinin was invented by Professor Yu Tu, who received the Nobel Prize in 2015. But gradually, what has happened? That the artemisinin-based therapies have started showing the resistance, and which is a big threat to the world. We have vaccine, but still they are not, effective. One of the vaccine is still under trial. Another vaccine, which was recommended in October 2021, it also requires multiple boosters, and just it gives a 30% efficacy, which is not effective enough. But still, it is recommended for this because we have nothing. It's better to have something. At least 30% efficacy will be given to the protection will be given to the children below 5. So at last, this slide summarizes that there is no effective single drug to treat the malaria. Unfortunately, after 1996, there is no drug appeared in the market. Now, you can imagine now why this drug discovery is so difficult, because it involves a lot of steps, and therefore it's a big pause, long pause, that almost more than 30 years, we have no drug appear to the market as effective single drug. In addition to that, during this pandemic, COVID-19, the whole focus of all the governments across the globe was to tackle the coronavirus disease, COVID-19, and certainly it has to be. But during this process, people started ignoring the neglected the other killer disease like malaria. And you will be surprised to know that in recent survey by WHO of 105 countries, 46 of the countries, they have reported the disruption in malaria diagnosis and even in the treatment, and which is a not a good sign. What will happen? The disease of malaria will spread more effectively. Now, there are various approaches which people adopt to find therapeutics. So I'm going to discuss two approaches. One is new medicines guided by biology, which seems to be very easy. Another would be new medicines guided by chemistry. So it is very easy. You identify a disease and make sometimes a library, being a chemist, and screen them, request a biologist, you get hits, lead optimization, make library of compounds, and you do a lot of target validation, protein ligand interactions, and in vivo, in vitro, whatever you do, and you get publications, and ultimately the biologist helped very well with the chemist, and it leads to a lot of outcome. But the bottleneck is how to optimize the leads for the preclinical and clinical studies. Example I give you very simple, as when I started my independent research career as a young scientist, there we synthesized a number of molecules, more than 80 molecules, and they were target based. So we utilized computational technique for the design of inhibitors against proteases, main proteases, and aspartic proteases of the malaria parasite, plasmapsins, plasmapsin 1, plasmapsin 2, and also plasmapsin 4. There we could trace out that there are a couple of molecules which have very good potency against all these aspartic proteases, and they are also able to kill the parasite in the culture. But gradually, the requirement has come up as that we need molecules. WHO suggested that we need molecules which are capable enough to kill the parasite during all the life stages of the parasite. Now, let me give you a little picture of how your life cycle of the parasite gets complete and why it is important to develop drugs to kill the parasite during all the stages. So you see, in the beginning, the infection starts in the liver. Then it goes to the blood. So in the liver, it is kind of a point where there is a minimum parasite load. And there gradually the parasite multiplies. And then it goes to the blood. Then within the blood stage, it has its own cycle. Then there is a gamete formation. Then there is a transmission stage, and then again that's how your sporozoite formation, and that's how your life cycle gets completed. So it is recommended now by WHO that we need to have drugs now which are capable to kill the parasite during liver stage and also blood stage. Subsequently, they are able to kill the gamete site formation, and also this transmission blocking stage pushes ukinate formation. And this scenario has started in last 10 years. So we were quite curious whether we will be able to develop such important molecule, and that will be called as multi-stage anti-malarial. Now, why it is required? Unfortunately, the current anti-malarial drugs, including artemisinin, they do not fulfill the requirement of renewed malaria eradication agenda by WHO because they selectively target only blood stage infection. Now, what could be the answer? So answer is that is there any existing drug that shows multi-stage activity? Dalways from UK, he took an initiative and did a lot of hard work in 2012, and he screened all the existing either approved or in clinical stages drugs, and he found that there are only two drugs, that is pyronaridine and atovaquone, which are capable to kill the parasite during all the life stages of the malaria parasite. But their efficacy is always compromised because of their cytotoxicity. So these both the drugs are not safe. And thus we can say that there is no single drug which can be called as multi-stage anti-malarial therapeutic. Then it becomes a place or the scope to work, all the chemists or biochemists work together to develop multi-stage anti-malarials to eradicate malaria if at all we want. So the journey started in 2017. A big collaborator of us, Professor Dominic, she started publishing and she invented a molecule which was very interesting, multi-stage anti-malaria. Also, gradually, Professor Kelly Chibel from South Africa, and also David Fedok, and she's Lynn Mary. She is also very good collaborator, all of us. And then we also entered into this area, and we published a very nice paper which has impact factor 12.99. And that's how we entered into this area, and we found a couple of molecules which were significantly able to kill the malaria parasite during all the life stages of the parasite. And you see one of the molecule number 5, seems to be very interesting, either in vitro or in vivo efficacy in blood stage as well as liver stage, and also gamete formation. Then we developed another library of molecules based on what we had achieved so far, and we developed novel anti-plasmodial compounds based on hydroxyethylamine. A very simple chemistry, you see, just simple ring opening anybody can do. Even a biologist can perform these reactions, and we found 4 of the compound hits which revealed their efficacy in sub-micromolar range. And the work was summarized and nicely completed and published in American Chemical Society Journal of Medicinal Chemistry, which is, I think, one of the premier journal of medicinal chemistry. So the work involves, you see, the 4 identified molecules. One of the top molecule, that is 5C and 5D. In particular, if I see here 5C, treated parasites did not grow beyond the late trophozoite stage. This is the another stage which is within the ring stage, blood stage, within the blood stage. And therefore, we had a feeling that the compound is capable to kill the blood stage, and also the compound evaluated in vivo animal cells, the animal models, in P-Burgei model, and there also compound showed good efficacy, both the compounds 5C and 5D. In particular, 5D was much better in animal models to kill the parasite in the blood stage. Then this slide is again very important. I showed in the beginning transmission blocking. So we were really curious to understand whether our compound is capable to kill the parasite and block the transmission stage. And interestingly, the compound showed very good efficacy. The 5D compound shown here is capable to block the growth of kidney development and also oocyst formation. This can be easily seen here. You see it is a control here. You see a lot of oocyst formation, and here in the treated, there is no formation, only just a couple of oocyst formation. And also here, it is also shown here also, you can see at 50 nanomolar and 150 nanomolar and also 450 nanomolar. So 450 nanomolar, almost it's over, and we were really very happy to know the result of this compound. And then we performed our computational strategy to understand what could be the possible structure activity landscape relationship index, and that is SALI, which we used open source platform. It is freely available. And we found that out of two compounds, 5C and 5D, they have a similarity almost 0.96 and being active in the asexual blood stage. We also did enzymatic assay. Before that, we did extensive in silico studies to find what could be the possible target of the compound, and we found that the compound probably targeting plasmapsin 10, which is the newly discovered protease, aspartic protease of the malaria parasite, and highly essential in during the growth of the malaria parasite. Then we did assay, and we found that compound has Ki value of 0.91 micromolars. So probably the compound is inhibiting the action of the plasmapsin 10 within the parasite, and that could be possible mode of mechanism. But certainly more additional experiments are need to be carried out before we conclude anything about the mode of action or target of the compound for any compound, in fact. Now, I come to another part of the presentation, that is new medicines guided by the chemical structure. So far, what we did, it was a kind of wonder for us because we synthesize a molecule and just give it to the biologist and request them, please screen them, and it's just like some time you get hit molecules. But now we become rational, and that's how a medicine chemist plays a crucial role. So here it's a very tough decision to find out what could be the possible pharmacophore to utilize, what could be the possible important scaffolds that can be utilized to build novel molecules. So we were also quite confused, but we could sense that yes, hydroxyethylamine, when it is clubbed in a single molecule along with piperazine, it gives a very good molecule, and that's how it was supported by the literature from Dominic's work. And from there, we took a lead and we found a very magical molecule that was just very simple chemistry. If you see, just lead optimization. Synthesis is just shown here. 3-step synthesis and very fantastic novel molecule, which was named as calixinin. Now, why it was named as calixinin, I'll tell you in the upcoming slides. It's a novel molecule developed by us, and it has been patented. Now, a US patent is filed, full patent, and you see the potency of the compound is 88 nanomolar IC50 and no toxicity almost on the liver cells. Then asexual blood stage activity of calixinin. So you see the last row, the compound showed against almost 90 nanomolar IC50. DD2, which is a DD2 strain of Plasmodium falciparum, which is a multi-drug resistant strain. And here also the compound showed 88 nanomolar IC50, which was really very encouraging for us. If you compare with chloroquine and dihydroartemisinin, these are the drug positive control that we utilized. Interesting point is here at the last is 50 nanomolar IC50 was noted for the clinical isolates which were received from Kenya. So it's a direct clinical sample, and this data is highly valuable, and that's how we got encouraged. The activity, activity of the compound was shown significantly in blood stage, and therefore we were wondering whether our compound is toxic or not in animal models. And we then conducted some experiments to assess the acute and subacute toxicity of the compound. And interestingly, the compound did not show any sign of apparent toxicity even at the dose of 1200 mg per kg oral dose. And this slide here at the right, you see it's a biochemical analysis of the serum, which seems to be almost normal with the ideal values, and the compound which was injected with our compound, the mice injected with our compound, almost there in the range. Then in vivo, this is in vivo data that I already discussed. The important part was the liver stage. Here I must tell you that, in fact, it's a repetition of my previous statement that in the liver stage, where the parasite starts growing, it has lowest percentage of the parasite, minimum load of the parasite. And that's why we need to, WHO suggested drugs to kill the parasite, vanish the parasite during liver stage, so that it will not lead to blood stage. Then it will not go to next, that is gametocyte or transmission blocking. So if you kill the parasite in liver, that itself is a sufficient or good strategy. If something remains, then multi-stage molecules, they will kill the parasite in blood stage, gamete side, and also the oocyst and okinete formation, that is the transmission blocking stage. And that was the logic behind the multi-stage antimalarials. So unfortunately, there are only few models are available for the liver stage infection assessment. If you want to do some assays, it's a little lengthy, tricky assays are available. That's why there is a logic that not many drugs have been developed exclusively to kill the parasite during liver stage. Our molecule is capable enough to kill the parasite at liver stage with the IC50 of 65 nanomolar. And also in liver stage in the mice model, the compound is capable to delay the infection by 30% at 10 mg per kg. Also transmission blocking. Yeah, here you will understand little better. Gamete formation, so it's 5 stages gamete formation, and our compound was significantly effective to stop this transition from gamete stage 3rd to 4th and 4th to 5th. Also, see, the gamete formation leads to the fertilization, then ookinete, then oocyst formation. So if you block the gamete formation itself, certainly you anticipate that, yeah, there will not be a zygote formation, and then ookinete and oocyst formation will not be there. Therefore, you block the transmission. And if you block the transmission, certainly your half of the job is done. Now, our compound was significantly capable to kill the gamete formation, mixed population, male and female both, and also it was effective to block the ookinete and oocyst formation at a different concentration, at particularly 150 nanomolar. Then we were wondering that, oh, what could be the possible target for this novel compound? Yes, here we find that the calcium channels are the possible target for our novel compound. And we did a huge, extensive in silico studies in collaboration with the University of Loyola Chicago and also Mayo Clinic, Florida, Jacksonville. And we found, yes, the compound seems to be target the calcium channels of the parasite. And that's how we named it as calixinin. This is a simple slide where we can see that the treatment of our compound at 500 nanomolar leads to a profound decrease in the intracellular calcium concentration in the Plasmodium falciparum trophozoites, schizones, and the ring stage. And this is a simple in vitro assays that we performed to see simple in vitro, you can say pharmacokinetics. And here we found that it has our compound did not induce any anticoagulant effect following the supplementation to the whole blood at the concentration up to 20 microgram per ml. So it did not impact on the clot formation. Now, this slide is again very important to compare the comparison of calixinin with the current antiparasitic and the compounds in the pipeline. So on the left side, if you see, it is the phase 2 and 3 compounds available in the literature, and this is our novel compound. So poorly understood mechanism of action, our compound now, by the time we are quite confident, yes, certainly there are some additional experiments in vivo that we are now still in the progress. And we found that, yes, it has a novel mechanism of action, a target. Higher dose required to treat. In our case, it's a nanomolar, you can see the data. Multi-stage activity, that is the big difference. Efficacy limited to single species. In our case, our compound, I did not show the data that our compound is very capable to kill the Leishmania parasite as well. Cross resistance to existing standard drugs due to the similar structure. In our case, our compound has unique substructure and target binding site. So there may not be possibility that it will have cross resistance. Lack of synergy with the standard antimalarials. In our case, the compound shows very good synergy with the known drugs, including the chloroquine and artemisinin. Complex secondary derivation from already expensive drugs. In our case, we can perform simple synthesis, inexpensive reactants, and scale up friendly. The compound has been patented, Indian as well as US patent, and we are now working towards the clinical stages of the compound. There are additional experiments, data have been received recently. I did not share because I thought it would be too much. Very simple slides I attempted so that it can be grasped easily by the students. So certainly I would like to thank all my collaborators here. At last, my mentor at MIT, postdoctoral mentor, and Professor Dominique here, you see she's a very good scientist, and Dr. Agam from NII, Professor Ravi Durasula from Mayo Clinic. And these are the very gem guys he's a wonderful virologist in the globe from Brazil. And she's a wonderful chemist, pharmacologist from University of Debrecen, Hungary, and the Charles Baisev Institute from Czech Republic. My group members, we have almost now 1.5 CR grants currently from BRICS and also DRDO in mass, bilateral grants, and lead optimization from CRG. And we have enough infrastructure in the lab now. And recently, I'm very happy to share that we are going to almost ready, and we'll just launch a BSL2 facility in the college, almost ready. We'll inaugurate it in the first week of June. So this is a short-term course which I conduct at University of Debrecen, Hungary, and CAPES Fellow at Brazil. And thank you so much for your patience and attention.
Thank you, Dr. Rathi. Now I would like to ask few questions that are with us. One question goes like, will it be possible for us to witness the end of the fight between humans and malaria?
Well, it's a, again, very typical question. Yes, it could be possible because you see, Europe is almost free from malaria. There is not a single case in last couple of years. If I am not wrong, almost 5 years, there is no case from Europe. So yes, it could be possible. Only thing is the proper management of the disease, transmission, what I said in the beginning, and also the therapeutics, they have to be effective.
One more question, that is it possible to develop a vaccine against malaria?
Well, for possibility, yes. I have to say yes. I cannot say no. There could be possibility, but it is not being developed because of many reasons. I told you, people understand like this degradation of hemoglobin. It's a very simple process. No, it is not at all. Nobody knows by the time. It's very complex process. Lot of enzymes. Daily people say daily they provide new reports that now this protease is essential, cysteine protease is essential. That means still there is a big scope to understand this simple biology. So yes, there could be possibility. People have been trying. Let's hope that it could be possible.
Yeah, there's one more question. Why are we not using the same management practices as in Europe to prevent malaria?
Oh yes, it depends. It depends. I believe that it will be gradually, I think, adopted. Here also, it needs to have very good infra, very good awareness, and there the management could be very easy. Lot of many reasons, you know that there could be so many reasons behind that. Certainly, those practices will have to come.
One more question is that do the Europe, which is not a tropical or subtropical country, they have cases of malaria? Because malaria is essentially a disease that is prevalent in tropical and subtropical regions.
Yeah, earlier there were a lot of cases, from Portugal and from other countries, if I remember very well, because I was in Portugal. There were a lot of cases, but gradually they have managed it so nicely. And certainly this is part of the like question. It has its answer. They have managed so nicely. Their management is so good, and the therapeutics. I think there could be many other reasons. Maybe we need to think more on it. So I don't know exactly what to answer on it.
Okay, sir. There's a question, interesting question though, that how can we do research with you?
So though I'm not, I was little very much, scary in the beginning when Dr. Ruchi just invited me, and because I already gave one lecture. So there were a couple of students after my this presentation last time you gave the opportunity. So they have completed their training in our lab, and they successfully completed, and they were very good students. So we have our group. We have some RAs in the lab. We have PhD students, and we have good collaboration, good funding also with the grace of God. Certainly, we'll be very happy. It is not that people will work with us. We'll be very happy to work with people, others. It's always to learn. It's peer learning. Yeah.
Yeah. And there's a related question though, that how can we pursue career in this field? So in general, I mean, if they want to, students want to pursue that, then what field they can choose or what trajectory they can use?
So they can go to any stream, doesn't matter, because in research, I did my PhD in totally something different, which has no connection with this medicinal chemistry. Let me tell you, I did on crystallography and all, some synthesis and totally something. But when I completed, I just forgot what I did. I initiated this because this was my interest piece of work, and I have been struggling. Though I have not done much, it's a simple. I'm a student, very small worker, learning in this area. It is always good to. Doesn't matter regardless of what you have learned in the past. Does not matter. What you want to do now, do it. You will have some problems, but people will be there with you if you want to do something.
Great,
As I
sir.
told you,
Great.
we are now we have developed DSL2 facility, culture facility, particularly for malaria parasite, leishmaniasis, and also there are some cancer cell lines. We'll be open it for everyone who wants to. Someone wants to do, they can come and do.
Great, sir. Great words of encouragement from you for the students as well as for us also. And now I would like to take this opportunity to thank you officially. It was indeed a great, as always, it is indeed a great pleasure to have you, Dr. Rathi, here. And it's really great to see the research work that is going on, that you are doing. And it's more so relevant to our country because the incidence of malaria and the deaths that are caused due to the malaria and dengue, they are really high. And it's commendable that your group is working on such an important issue that is more pertinent to our country. So I really thank you for sparing your time. I know you had a very busy, schedule, as you are actively involved in this research and many more things. So I really thank you for sparing your time and making it possible to come here and give this very informative and very insightful talk.
Thank you,
Sir.
ma'am. Thank you so much for the opportunity, and it is always pleasure to meet our own colleagues. Of course, it would have been better if you meet,
In
offline.
offline.
Yeah.
Yeah.
So
Surely
that
we would like to have you in our college for a talk, and we will really seeing an opportunity to invite you here.
oh, ma'am, thank you so much, and so kind of you. And again, congratulations to all of you. You have been doing really good job. Thank you so much, ma'am. Thank you.
Thanks.
Have a nice
Thanks.
day. Thank you all.
Same to you, sir. Now, coming up next is our poster and oral presentation session. And I would like to tell you that these two sessions are going to operate parallel. At the same time, simultaneously, they will be conducted for the oral presentation. The same link, the same meeting will be used for the oral presentation, while for the poster presentation, there is a link that will be just shared on the chat box and as well as on the Telegram for the poster presentation. So all the participants who are there for the poster presentation are and those and all other audience that wish to join the poster presentation session, they are requested to join another link, another meeting with the help of the link. And after the poster session is over, they are requested to join back in the this present meeting. So, welcome back, everyone. We are going to start with our oral presentation session. And for that, we have our judges with you. So I would like to introduce the judges. First, we have Professor Sandeep Das with us. So can you please open the camera? Welcome, sir. Sir, you are muted. Good morning, sir. Good morning, and I welcome you to this oral session, oral presentation session. And Professor Das, we already had the opportunity to hear him in the inaugural session, and he is professor at Department of Botany, University of Delhi, and his area of research is in genomic studies in members of Brassicaceae, then evolution of plant morphological diversity and adaptive variations. Then he has diverse area of interest in his research, which includes role of miRNA and transcription factor, then impact of polypolarization on regulatory genes, and so on. So we welcome you, sir, here. The next judge is Dr. Babita C. Kola. Ma'am, can we hear? Can you open the camera? Yeah, please. And we have Babita ma'am with us, Dr. Babita C. Kola. She is currently Associate Professor in Zakir Hussain Delhi College, University of Delhi. She has earned her PhD degree from University of Delhi, and her area of interest are genetics, molecular biology, and plant systematics. We welcome you here, ma'am. And we have a list of participants who are there for the oral presentation. I'll be calling their names one by one, and the presenting author has to present for 5 minutes, and 2 minutes are there for the question and answers from the judges as well as from the audience. So we can start with the session, ma'am, sir.
Yeah, it's fine. Yes.
Yeah. Just a second.
Can you tell the rules, explain the rules to the participants, please?
Okay, the rules are they're very simple, that your presentation has to be on the PowerPoint mode, and it has to be of 5 minutes, and plus 2 minutes are there for the question and answer. So you please take care that you do not exceed the time limit that is for the 5 minutes. And there will be one best presentation that will be adjudged by the judges, our esteemed judges, and the judges' decision will be final. So we call upon for the first presentation for oral presentation, that is in silico molecular docking and pharmacokinetics of selected phytochemicals against BK channels. Archana Raj and Suresh N. Nair. I think Archana Raj is the presenter. I mean, the presenting author is Archana Raj. Can we have Archana?
And Dr. Tabassum, who's keeping track of time?
I'll keep, ma'am.
So, Dr. Tabassum,
Yes, sir.
just one more point is, I think when they are presenting, you could also let us know whether from where are they, their affiliation, and if they are
Okay.
PhD student and or are they scientist, if you have that.
Right now, I don't have that in consolidated. So maybe the author, the presenter, they will themselves introduce them and the affiliations. So.
Okay. Okay.
Fine. Can we have Archana? Archana Raj? Okay, meanwhile, we can go to the next one. Redesign of natural proteins of novel applications. That's Rashi Sharma and Tia Verma.
Excuse me, ma'am. Excuse me. Sorry to interrupt, but the first presenter is there, and she has written something in Telegram group. Maybe she is not able to join or not
Okay.
able to connect properly.
The first presenter, that is Archana Raj.
Yes, she has written message in Telegram group.
Okay, we'll just, Dr. Savinder, can you just see that? Dr. Savinder? Or Dr. Vatal? Okay, we will just see
Check.
what's it. Yes, ma'am.
She's asking, has the oral presentation started first? That means she's not able to connect. We'll take her later.
Yeah, we can take her later. Yeah.
Right.
Then for the second presentation, Rashi Sharma, is she there?
Yes, ma'am.
Yeah.
Ma'am, am I audible?
Yeah, you are audible.
I'll just share my screen if you can confirm.
Yeah, you can. Yes, you can. Meanwhile, you can introduce yourself, your affiliation, and your co-author's affiliation.
Sure, ma'am. I have that written in my presentation. I'll just do it.
Okay, fine.
Is the screen visible?
Yeah,
Yes.
it is visible.
Okay, so I'll start in 3, 2, 1. Warm greetings to all the listeners. I am Rashi Sharma along with my co-author Tia Verma. We are from Delhi Technological University and we are here to present our overview of a work titled Redesigning of Natural Proteins, that is enzymes for novel applications. So moving on, proteins are linear polymers of amino acids who perform manifold roles in cell pathology. Protein structures is knowledge obtained from various branches of biology, including biochemistry, biophysics, structural and molecular biology, bioinformatics, and computational biology. This has generated curiosity in researchers to redesign and engineer these proteins with destined functions to improve whatever we have got from the nature naturally. Enzymes have several industrial applications, like for starch processing, we use amylase. For textiles, we use cellulase, catalase. For detergents, protease and lipase are known enzymes. And similarly, for food and pharmaceuticals, for bioremediations, enzymes are used to degrade xenobiotic compounds, and this usage of enzymes and their wide adaptability has led to researchers aim to redesign and make new usage of the for novel applications. Moving ahead to talk about what we have brought in our work, the recent advancements have brought two approaches to design these synthetic proteins. First is the experimental or molecular methods that are based under direct evolution, and second are the computational approaches or in silico or proteomics computing techniques that are being used to create these to redesign these proteins. We have proposed a comprehensive overview in our work for proteomics tools to redesign enzymes. Although both the approaches have their pros and cons, they complement each other, and we have also proposed an semi computational approach where we have where we show the verification of our computational data through experiments. So this is the basic thing. Computational designing has two major approaches: the rational approach or the de novo designing approach. Computational approach uses bioinformatics and two molecular modeling techniques that have been trending to make the protein desirable structure and functional attributions. Here in the table, we have summarized some of the enzymes belonging to various class or functionalities that have been designed differentially from differential techniques. Here, the DE stands for direct evolution, that has used the molecular or experimental approach. CD stands for computational design, and DN is for de novo design. So, for example, the organophosphate hydrolase has been redesigned using the computational tool Rosetta. So, as we have told that there are two approaches, now we'll talk about the first approach, that is rational designing. So for comprehensive structural and functional information, we essentially need the sequence or the code of the protein. So these are the methods that have been developed to extract these essential informations for structure and function related relationship. Under this, we have covered three major techniques: multiple sequence alignments, co-evolutionary analysis, and third being the structural prediction methods. This is a infographic showing the detail about all the four methods. Later on, talking about MSA, this is a simple way to analyze the unknown protein and compare its alignments in sequence to a template protein. The several tools that are known are MFFT K-align, Prob-align, and other. These are gaining vast importance in bioinformatics for sequence analysis. Secondly, we have co-evolutionary analysis. This phenomena occurs via a pair of residues, and these present a distant view of the sequence rather than proximity view. And this was used in CGAK, asparatokinase, and this is recently being used for new 3D fold to design the 3D folds of proteins. Moving on, we have summarized the computational tools, algorithms, servers available freely for protein designing along with their uses. These are some of the things we propose to elaborate. Moving on the structural basis design, we have 4 techniques under this, and since we do not have time to explain on of these 4, namely are
Time is over.
emergent modeling. Thank you, ma'am.
Thank you. We can go for questions. If the judges want to ask any question from the participant.
I would request Dr. Professor Das to go ahead with the interaction because it is his area of expertise.
Good morning, Rashi.
Good morning, sir.
So just wanted to know, you are a PhD student or which year?
Sir, I'm undergraduate second year student.
So whatever you presented, have you done any experimental have you done any analysis on your own to, because your title says redesigning of proteins for novel function.
Yes, sir.
Or are you going to do sort of de novo? Because what is this de novo approach that you are talking about?
Sir, to answer the first question, we have not done major of the work in the redesigning part. We are reading these algorithms and these softwares. Basically, we use this opportunity to get acquainted with the available resources. We have started out with doing um, molecular docking, and then we are working to learn stimulations. Apart from that, we are learning machine learning separate. We have used Bayesian's theorem and separate things, but we haven't actually established as we propose de novo or new enzymes by the methods we are showing. We presented this as an overview to let people know of the sources available through which redesigning can be done.
So just one last quick question. Have you heard of 1D, 3D principles of protein structure? And since you talked of machine learning or something, I believe there are training sets that are used in machine learning. So would that not bias the approach towards existing protein structures and existing protein functions? So where does the novelty come from?
Sir, we are proposing to feed the machine with a lot of various exceptional data also. We are not just making it available to the usual data. We know that there are some biases in the machine related to experimental approach, which is completely independent. But we propose to do an elaborate research of different 1D, 2D matrices.
Thank you, Rashi.
Thank you, sir.
Any question from the audience?
One small question, Rashi.
Yes,
it was a good presentation. How did you identify paired residues of significance?
Identify the residues of significance.
Yeah.
Ma'am, the active residues that are available on the databases that told that these active bases are responsible for catalytic actions or folds or functionality of the proteins. We targeted those proteins, we marked them using the softwares, and then we targeted them.
Okay, thank you.
Thank you, ma'am.
Thank you, Rashi.
Thank you, ma'am. Can I stop presenting?
Yeah, please. The next presenter is Abhishek Roy on omics-centric approaches to classify bacteria. Abhishek, can we have you here?
Good morning, ma'am. Good morning, sir. Yes, ma'am, I can hear you.
Yeah.
Is my voice audible?
Yeah, it's audible.
Good morning, sir. Good morning, everybody. Good morning, ma'am. My name is Dr. Abhishek Roy, and I am presently working as a faculty in Vivekananda Institute of Technology. and they have a branch called Vivekananda Global University. So I'm a faculty member at Vivekananda Global University, which is in Jaipur. And I'm teaching BSc MLT students, where MLT stands for Medical Laboratory Technology. And though I'm not very well versed in the field of proteomics, I thought of getting some knowledge by attending this conference. So whatever I'm presenting, it is not my work. I've read a few papers and I'm trying to summarize the brief research work that has been done in the context of bacteriology. So since I'm teaching students about pathology also, and we are teaching them about certain pathogenic bacteria like Streptococcus, Staphylococci, etc. Let me start the presentation. I'll show you what I'm trying to say. Please give me a minute. So basically, I'm trying to talk about the application of proteomics and bacteriology, wherein, as it is well known, that proteomics could be very well applied in understanding the expression levels of different proteins that are critically important for the pathogenicity or the pathology of the bacterium. So today I'll be focusing on Streptococcus. So I'd like to focus on some proteins of Streptococcus that by proteomics have been targeted for being down-regulated to use as a therapeutic measure. Starting off with what is a proteome, as it is well known, a proteome is basically a collection of all the proteins of a cell. There are different modifications, the post-transition modifications like sulfidation, ubiquitination, methylation, nitrosylation, etc. And we have abundant databases right now present in this context. Like we have a very important database called MOPED. So MOPED stands for Model Organism Proteome Database. And we have certain consortiums as well, like Proteome Exchange Consortium. And basically the purpose of all such databases or aggregations is to promote the interactivity among the databases of the humongous number of proteins that are present, so as to understand their functions and interactive networks. The challenges of proteomics is that the data is quite complex, considering the fact that proteins undergo a number of post-transition modifications or PTMs. Also, proteins undergo degradation. Moreover, we do see the phenomenon of alternative splicing. So from the same transcript, probably different number of polypeptides could be generated by alternative splicing. These all contribute to the complexity of the data. But the biggest advantage perhaps of proteomics is that the protein expression, as we can see on the last line of the slide, the protein expression during an infection can be very well observed. Meaning to say that if there are some proteins that are down-regulated or up-regulated during the course of infection by a particular bacterium, if we come to know certain proteins that are down-regulated or up-regulated during infection, we can probably carry out more research on them and try to target them. Focusing on a particular bacterium called S. mutans or Streptococcus mutans. Now, as you know, Streptococcus mutans is a very dangerous pathogen. It exists in the mouth of a normal human being, but it is known to cause severe damage to the teeth. As it is known, Streptococcus mutans can damage the enamel of the teeth, and it carries out production of acids like malic acid and lactic acid, which go on to further degrade the enamel and then cause the disease called dental caries. Now, in this regard, some research has been carried out by scientists wherein they have focused on a particular protein of Streptococcus mutans called SMU833. Basically, SMU833 is a glycosyl transferase, and it plays a critical role in the biofilm formation and the growth of Streptococcus on the human teeth. Now, when they deleted this particular gene, they, with the help of proteomics, try to identify which are the proteins, the other proteins that are affected or are downregulated thereby causing hindrance in the growth or infection caused by Streptococcus. And they went on to observe that there were certain proteins which are involved in promoting resistance to oxidative stress. For example, we have thioredoxin reductase. We also have certain proteins called GTFB, GTFC, GTFT. These are basically, again, glucosyl transferases. So number of such proteins are found to be down-regulated, so as to say their expression levels became quite low once SMU-833 was deleted. And thereby the conclusion was that once SMU-833 is deleted, some other proteins are impeded in production, which eventually results in lack of resistance to oxidative stress. Now we know that resistance to oxidative stress is necessary because in our mouth, not only Streptococcus mutans is there are a number of other bacteria that are producing hydrogen peroxide all the time. Now, to combat and to compete with the other bacteria present in the mouth, Streptococcus mutans can very well, combat the oxidative stress produced by the other bacteria in the mouth, thereby promoting its own survival. But if you delete SMU-833 the gene, then the other proteins that are responsible for contributing to resistance to oxidative stress will be depleted, and thereby S mutants cannot survive much. Also, it was some more research has been carried out wherein they have administered an antibiotic called bacitracin. So this is an example wherein we deliberately induce a stress response in the bacterium. And after the stress response is elicited, we try to observe what are the other expression levels of the other proteins. And there also they found certain proteins in all, at least 12 proteins are identified that were down-regulated when bacitracin antibiotic was administered. The challenge is we need to identify the proteins, but what is the source? Like, how can we, identify the proteins which are upregulated, downregulated? So when it comes to the human context, in the human body, if we want to identify certain proteins, then the body fluids can be taken and they can be analyzed by liquid chromatography, mass spectrometry. And in the present date, one of the most important human sample or body fluids that are taken is BAL, that is bronchoalveolar lavage. And of course, blood is another very good sample. But
time is up.
Yes, ma'am, just one more minute. When we take blood, the disadvantage is that, there is the masking of the low copy number proteins by the high copy number proteins. So at least there are 15 proteins in the human body that are abundant, and they occupy approximately 70% of the volume or the content or the concentration. So the proteins that are abundant, for example, hemoglobin, etc., they would of course mask the other low copy number proteins, and we might not identify the low copy number proteins. That's one disadvantage. Thank you. This was one more slide for subtractive proteomics, but I'm sorry, due to lack of time, I'm not speaking. Thank you.
Thank you, Dr. Abhishek.
Good morning, Dr. Abhishek.
Morning, sir.
So I have one small query. You talked of this SMU-833. This is a protein or a gene that encodes a protein from the pathogenic bacteria that you're talking about.
Pardon, sir. Please repeat. I'm sorry. You're talking if it's a protein or not?
No. This is a protein that is encoded by the bacterial genome, right?
Yes, sir.
And so is it a secretory type of protein? I mean, in the sense of why are you looking at the BAL, the alveolar, this thing, because is it secreted into the human, this thing? And I believe you talked of something about the dental.
Yes, this particular protein contributes to dental caries, the disease of the human teeth. And since it's a glucosyl transferase, it helps in acquiring, in the metabolism of glucose. Of rhamnose, sugar. So it is contributing to the metabolism of sugars by Streptococcus and promoting its ability to carry out dental caries.
But the protein is, how is the protein found in the human, the BAL is a human fluid?
No, sir. The protein is not found in the human body fluid. It has been extracted. It is found in the bacterial culture itself. I was just trying to extend my presentation by talking about other samples. Apart from bacteria, what are the other samples that you can take during human infection to analyze protein?
Okay.
But SM is taken from the bacterial culture itself.
Bacterial culture. Okay. Thank you.
Welcome.
Some more questions? Okay, thank you. Thank you,
Thank
Dr.
you.
Abhishek. You may
Thank
stop presenting. The next,
Thank you. Thank you.
the next speaker is Bhani Kongkang. Are you there?
Yeah, ma'am, am I audible?
Yeah, you are audible. And her title is, just a second. Her title is Computational Studies in to evaluate the SHV1 beta lactamase inhibition ability of Ficus religiosa secondary metabolites. So over to you, Bhani.
Ma'am, should I share my slides?
Yeah, please. And you can introduce Abhishek. Can you please stop sharing? Thank you. Yeah, Bhani, you can share your slide and introduce yourself, please, and your co-author.
Yeah, just a second, ma'am. Ma'am, is it visible?
Yes, it is visible.
Good morning, everybody. I am a research scholar in Environmental Biotechnology Lab of Delhi. And the topic of my presentation today is computational studies to evaluate the SHV1 beta lactamase inhibition ability of Ficus religiosa secondary metabolites. Coming to the introduction portion, as we know that antibiotic resistance is a major threat to the humanity. Since the discovery of penicillin in the year 1927, there was a massive reduction in the number of bacterial infections. However, in due course of time, a number of bacteria became resistant to the existing antibiotics. As a result of which, right now, the pipeline of antibiotic is gradually diminishing. And according to a WHO report, if the current trend continues, nearly 10 million deaths will happen by the year 2050. Now, beta lactams are one of the most widely used antibiotics. Nearly 60 to 70% of the antibiotic used in the world consists of beta lactams. Now, beta lactams are the class of antibiotics that have a beta lactam ring structure, which is the pharmacophore of this class of antibiotics. And they are divided into four major classes, which is the penicillin, cephalosporin, carbapenem, and monobactam on the basis of their structure. Now, resistance to beta lactams can be developed by many mechanisms, but the major mechanism is by the production of an enzyme known as beta lactamases. Now, beta lactamases are basically hydrolases, and they are divided into two parts, that is, two types on the basis of serine and metallo. On the active side, if you have a serine residue, it is classified as class A, C, and D. On the active side, if you have a metal residue like a zinc plus iron, it is a class B beta lactamase. Now, these are the representative structures of the beta lactamase. SHV1 belong to class A, and imipenem 1, which is the metallo beta lactamase, has a zinc ion on the active side. So these different classes of beta lactamases, they have different substrate affinities, they have different ability to hydrolyze the beta lactam antibiotics. Keeping this in mind, I have designed my study to identify some sort of inhibitors for this beta lactamase enzymes. So, for my in vitro assay, I have screened 30 plant extracts in my lab for the ability to inhibit beta lactamase. I have also checked the ability of this extract to potentiate beta lactams. However, this is not a part of my presentation because of the limitation of the time. I have just focused on the molecular interaction that we have done to understand the binding of the metabolites which is present in the active extract with a standard beta lactamase, which is SHV1 beta lactamase. So what we have done is, after performing the in vitro assay, we narrowed down to one plant extract, which is the Ficus religiosa bark acetone extract, and we have done the HRLCMS analysis of this particular extract to understand the composition of the metabolites that is present. And finally, we have identified one protein, which is SHV1 beta lactamase, which is one of the most commonly predominantly found beta lactamase, and we have performed the ensemble docking with this particular protein using the Autodoc Vina module of Pyrex. And further, to understand the molecular interaction, we have done the MD simulation for 50 nanoseconds at the high computing facility of IIT Delhi. Now coming to the result portion, from the ensemble docking, we found that we have actually performed the ensemble docking of 200 metabolites with SHB1 protein. Out of that, the top metabolites were quercetin, myrcetine, leutonine, texifolin, and meclianin, which were having a binding energy of -5, of -7, less than -7, and this were better than the binding energy of avivectin. Avivectin is a standard beta-lactamase inhibitor which is currently available. Now we visualize the meclianin interaction with SHV1 by using the Discovery Studio. This is a 2D interaction. This is the meclianin. It has a glycoside bond. So this is a typical flavonoid structure, but it has a glycosidic bond here, and it was found to interact with some of the important amino acids like serine 70, serine 130, which is present in the active site, as well as the neighboring amino acids of the enzyme. Now, to understand the molecular interaction, we have done the MD simulation. The data of the MD simulation, which was done for 15 nanoseconds, were first validated by examining the RMSD, RMSF, RG, and SASA. We found that during the course of the simulation, the RMSD, as we can see, got stabilized, indicating that the protein was equilibrated. And at the same time, the introduction of ligand like avibactam and meclianin did not alter the configuration of SHB1, which is indicated by less fluctuation in RG as well as SASA and RMSF. Now, to understand the strength of the binding of the standard ligand as well as the plant metabolite, we also checked the number of hydrogen bond which was formed during the course of simulation and the hydrogen bond occupancy. We found that a fair number of hydrogen bond number was found, the maximum number of which was 6. And in terms of hydrogen bond occupancy, we found that meclianin was forming a very good hydrogen bond of more than 150 hydrogen bond occupancy with GLU 240, which is not in the active site, but which is not in the active site, but which is present around the active site. Okay, we also wanted to know that during the course of simulation, whether the ligand which is presented in cyan color here is expelled out from the active pocket or not. So we visualize the different frames during the course of simulation. So this is the 0 frame, and we visualize till the 50 nanoseconds. We found that the ligand is in the active pocket of the enzyme only, but the main interesting thing is that irrespective of the ligand orientation at the beginning of the simulation, when it stabilizes, we found that the glucose moiety, the glycoside moiety, was interacting towards the omega loop. So basically the beta lactamase has different motifs which are conserved. So this particular glucose moiety was found to be oriented towards the omega loop of the beta lactamase, and this red color are the active pockets. We also found hydrogen bond interactions with amino acid residues which are neighboring, for example L-alanine 237, which is neighboring the active site but not exactly present in the active site. Now we also analyzed the binding energy of the standard as well as the meclianin by using the MMBPSA calculation protocol of Rashmi Kumari et al. And we found that the binding energy of meclianin was comparatively better than that of the avibactam, indicating its affinity. So the major conclusions of my study is that from the in vitro experiment, we found that Ficus religiosa is the potent plant. Then the metabolites were docked and as well as simulated with SHB1 protein. We found that the meclianin has the ability to inhibit SHB1 protein by binding with basically with the amino acid
Time is
residues,
up.
which is neighboring the SHB1. And because of this, by blocking the enzyme, not the active site, but the neighboring areas, they may inhibit the activity of the enzyme, but however, we need to purify the active compound for further analysis. This is my references. Thank you. I'm open to any questions if anybody wants to.
So Bhani,
Yes,
can you
sir.
hear me?
Yes, I can hear you.
So nice work. So are you in 3rd year, 4th year, 5th year of your PhD?
Sir, right now I'm in my 3rd year of my PhD.
And this is your PhD work?
Yeah, this is a part of my PhD work. We have done a lot on the in vitro side as well, but because of the time frame of the presentation and the molecular drug design, I have just narrowed down to the in silico portion that we have done.
No, that's okay. So the compound that you got is from the bark of Ficus religiosa, right?
Yes, sir. We have actually screened around 30 plants. From there, we narrow down to this plant.
Okay, no problem. So within Ficus also, do you know the tree from which the bark was? I mean, what is the age of the tree, and what all other tissues have you looked at in within that same Ficus?
Yes, sir. Actually, the tree is located in IIT Delhi itself. We have taken the bark as well as we have taken the twig as well as the leaves portion, but we could not find a similar kind of activity in terms of the in vitro assays. In when we come to leaves and the twigs, so we found the activity in the bark, and similarly for other plants also the beta lactamase inhibition we are mostly finding in the root portion of the plants as well as in the bark portion. So we have just carried ahead our experiments with the bark, not with the other parts.
Okay, so apart from the in vitro assay that you did, I believe you must have used some beta lactam class of
Yeah.
antibiotics to.
Yeah, sir. We have also done the microbial assays to see whether our extract is able to potentiate the beta lactam. We have used the ampicillin, which is the basic beta lactam. From there also we found that this extract was able to potentiate the beta-lactam ampicillin by reducing the IC50 against a number of resistant bacterial strains like Pseudomonas aeruginosa, Streptococcus aureus. So from that only we have narrowed down and we have performed the molecular interaction just to see if at all it is binding, where exactly will be binding. Will it be inhibiting the active site or will it be blocking the active site so as to prevent the substrate binding to the beta-lactamase? And here, because of the maybe because of the presence of the glucose moiety or the larger size of the meglianin, it was found that it was actually blocking the active site, but not actually binding to the active site. And it's blocking the active site, then maybe the substrate will not be able to bind to the active site, as a result of which a substrate like ampicillin will not be hydrolyzed.
Okay, thank you. Thank you.
Thank you, sir. Thanks. Thank you, ma'am.
Thank you, Bhani.
Hello, Vani.
Yeah, ma'am.
You have mentioned a term PSM. What is that?
Ma'am, actually, it means plant secondary metabolite. It was just a short form of the plant secondary metabolites.
Okay. A small question for you. Did you try any ADMET analysis for metabolites?
Yes, yes. After the in silico molecular docking studies, we have also done ATME in terms of Swiss ATME as well as PKCSM, which are the softwares which are available. We found that our this plant compounds were not toxic as compared to, they are not hepatotoxic as compared to the standard drugs like a clavulanic or a avibactam as of now. And it was also found to be very good in terms of viability as per the studies, in silico studies. And right
Thank
now we are focusing on purification of these compounds, which will
that.
later on be done on a cell line. Yeah.
That. Thank you.
Thank you, ma'am. Thank you.
Thank you, Bhani. You
Thank you,
may stop presenting your slide. The next presenter with us is Misha Yadav, Dr. Misha Yadav. And her topic is foreign protein production using oil body membrane proteins. Hello, Misha. So
just give me a moment, please.
Yeah.
Am I audible enough?
Yeah.
Okay. Hi, good morning to the judges, to the organizers, and to the audience. I am Dr. Misha Yadav, currently working in Department of Botany, Shivaji College, and along with my co-author, Dr. Sandeepa Singh, I would be talking about, who is faculty from Department of Botany, Day3 College. I will be talking about foreign protein production using oil body membrane proteins. So during my PhD, I have worked with oil bodies and oil body membrane proteins, but this presentation is beyond what I have worked in lab, and this is a review article as such. In this presentation, I would be talking about oil bodies and oleogens and how they can be put to use for different biotechnological purposes. So before starting further, I would like you to know what are oil bodies. These are spherical organelles, and mostly they are abundantly found in oil seeds. In other tissues also, they are present. And if you see them under microscope, this is a picture, an image where you can see these white structures. Just a second. These white structures are oil bodies, and if you stain them with any fluorescent dye such as Nile red, you would find them like this. These pictures are from my research work only. If you see a cross-sectional view of the oil bodies, this is how they look. The central core is made up of triacyl glycerols, followed by a membrane, and this membrane is made up of phospholipids. This is a monolayer. It bounds the matrix or the core, and on the surface of this membrane are bound specific proteins such as oleosins. When you see the structure of oleosins, you'll find that they have an N and C termini, which are exposed on the cytosolic side. Whereas there is another domain which is highly conserved. It is spanning across the phospholipid membrane, and the oleosins are like a part of it is exposed on the other side, that is the matrix side of the oil body. So this is how it looks. So this is the N terminal, the C terminal, and the central conserved hydrophobic domain. Because of the peculiar structure and the properties of oleosins, they have been put to different uses. Many of the uses include their use as emulsifying agents. They have been used as carriers of recombinant proteins, as well as immobilization matrices. And not only the natural oil bodies have been utilized, oil bodies can be created artificially in labs, and such oil bodies are known as artificial oil bodies or AOBs. And these AOBs have been again put to use for various purposes, such as for purification, refolding, and immobilization of recombinant proteins, and for other purposes as well. So this is how the process is carried out. The oleosin gene is fused with the foreign gene or the gene of interest, and then it is allowed to transcribed and translated. And upon translation, along with the oleosin, the gene of interest or the foreign gene is targeted onto the surface of the oil body. And once it is targeted to the surface of oil bodies, we can see this in this picture. This has been taken from one of my publications. So this is how the sequence is created. The promoter is there. This blue one is the oleosin gene, then a linker peptide followed by the gene of interest. And here upon like transcription and translation, you will find that the oil body is carrying the oleosin along with the protein, which has been the gene for which has been incorporated here. So these oil bodies, once they have been formed, they can be used either directly with the protein still onto it, and they serve as immobilization metrics, and expression of proteins such as xylanases has been done using this type of methodology. In other cases, the protein is cleaved from here, and the protein is then separated, purified, and used for different purposes. For example, in case of hirudin, the second methodology was adopted. This slide shows further elaboration of what I have discussed, but I'm skipping it for now. Then comes the why do we prefer these organelles over other methods? The thing is, it is very easy to isolate such oil bodies, and we can carry out simple processes in labs, such as like you just homogenize the material, then centrifuge it, and upon washing, you will get a clear layer of oil bodies on the top of your append of, or if you are using some bigger like glassware. And in this picture, you can see how it looks. You are going to get a clear cut layer, and the process can be carried out in lab easily, as well as for industrial purposes or on larger scales. You can carry it out like this, and you can use the purified oil bodies or the purified protein for further usages. Now, coming to artificial oil bodies, earlier it was like this was a limitation that one needs to grow the entire plant and wait for the oil bodies to be generated in the seeds, and then one would be able to like isolate the protein. But that limitation has been overcome by artificial oil bodies, and these can be easily created in lab using triacyl glycerols, phospholipids, and proteins such as oleosins, which are the membrane proteins in oil bodies. Sonication is carried out, and the artificial oil bodies are created. These are a couple of pictures. the benefit is that when we use artificial oil bodies, we can easily purify, immobilize, and refolding of recombinant proteins can be done even if the protein is produced in system apart from plant. For example, in bacteria, if you are expressing some gene and you have got the protein, it can be further transferred onto the oil body by generating artificial oil bodies. In the recent times, oil bodies have been used as emulsifying agents, and they have also been used to aid drug delivery. And recently, okay, just give me a moment. So there have been a couple of applications. It has been used as drug delivery system for cancer drugs as well as for other purposes such as for human fibroblast growth factor and for preparation of different enzymes such as xylanases and other proteins. Thank you for your time and attention.
It's open to questions now.
So I have one small question, Dr. Misha.
Yes, sir.
So I left in between because my connection got lost. But I believe you're talking of fusing the oleosine protein with other proteins that are, sort of being to be packaged within the oleosine as a transport body. So
Yes,
is
sir.
there a limitation to that strategy based on your knowledge, your experience, and how would you overcome that?
So earlier, a huge limitation was if you carry out this process in plants, the oil bodies are abundant in oil seeds. So one would need to wait for an entire cycle for the seed to develop, and then only abundant oil bodies could be isolated. But nowadays, that limitation has been overcome by formation of artificial oil bodies, which can be done in lab easily. So that is one of the limitations. And another limitation is when we are using these oil bodies for different purposes, sometimes people are allergic to the oil bodies of a particular type. So that needs to be taken into account. And there are some medical.
Is there a limitation the oil?
Sir, I'm sorry, I'm not able to hear.
Please unmute yourself. Actually, there was background noise, so I have muted all.
Okay, ma'am, I have unmuted.
Yeah.
Sir, can you please repeat your question?
I think he's got disconnected. Any more questions? Okay, thank you, Dr. Misha.
Thank you, ma'am.
Yeah. The next presenter with us is Monica Mishra, along with the co-author Bhumika Chauhan. And the title of their abstract is Genomics, a deeper insight into the biology of gene expression analysis of a digenetic nematode parasite. Yes, ma'am.
Can
Can you adjust your camera a bit?
Yeah. Am I visible now?
Now it's
Better?
Yeah.
Can you see my presentation?
No, it's not there. Yeah, now it's there.
Where? Sharing. Okay, sharing was paused for a moment. Yeah. Now is it there?
Yeah, it is there. It is there.
Right. Thank you. Thank you so much.
Yeah.
Good morning. I think it's still good morning. Good morning, everybody. I'm myself, Dr. Monica Mishra, Associate Professor from the Department of Zoology, Acharya Narendra Dev College, Delhi University. The paper that I am presenting today is the first report of insights into the motif of the trematode parasite that I'm going to talk about.
and a deeper insight into the biology of the gene expression analysis of this parasite. It has already been published. Some part of it I'm going to present today. I go to the next slide. Basically, we talk about omics technology. That is, when we say omics, we mean genomics with the DNA and transcriptomics with the RNA being transcribed and the proteomics to the protein. I will not go into details. We are all aware of this central dogma. Now I go to the next slide. Courtesy. This slide is courtesy Professor Sandeep Das, whom we listened on the first day. He explained in detail all these points. Basically, he explained on the system of plants. Now, what I talk about right now is animal system where agriculture will be replaced by medicine and crop improvement is replaced by disease management, right? So this was detailed today. Medicine and disease management has been very well dealt today by Dr. Vinay, both by Dr. Vinay and Dr. Brijesh Rathi in the morning. But basically today I will be concentrating on the disease, that is wet disease on the buffaloes, which with the model that we have. So again, this is another slide, courtesy Professor Sandeep Das. What he tried to say was that the knowledge and understanding of genomic organization and function in the plant breeding was through these technologies and finally going as a knowledge. Now, what we do in the zoology system is this, that we go to the next slide to see this. The slide that shows here is genomics, where we are able to screen and diagnose newborn screening, prenatal testing, disease susceptibility, and then therapeutic decisions we are able to take, as well as monitoring the disease burden by relieving the need to have animals as the protocols. So I go to the next slide. This is the integrated omics that we had yesterday learnt about from the, again from the Department of Botany DU, Professor Renu had in detail talked about all the interplay of proteomics and metabolomics, especially proteomics she talked about. And today, what I'm concentrating on is this slide. That is the parasite that I'm going to talk about is Explanatum explanatum. This parasite is a digenetic trematode parasite found commonly in the bile ducts and gallbladder of cattle and buffaloes worldwide, whereas in Asian countries, it has been reported even in goats. Recently, one paper has come up where even in goat, this has been reported. Now, this produces productivity loss. How does it do so? It produces a disease called as amphiosteomyces. So amphiosteomyces is a disease which actually this is actually the picture of the parasite that we are seeing right now. This parasite goes into the bile duct of the buffalo liver. Adult worms remain in here and they have an intermediate host, that is species of snail from the genus Limnae. When the eggs are discharged along with the feces of the host, that is the buffaloes, these free-swimming larvae will hatch out from the egg, will penetrate the snails, and the specific genus from these snails will now undergo larval development. They will emerge out of it, swim freely, and will become encysted on the vegetation. So they will remain there in the encysted state of metacercaria, and they will be finally ingested by the final host, where they will go into the intestine, migrate to the surface of liver, and finally reach the bile ducts. Many times they cause the death also of the cattle. So it's a very, very a disease which has taken lives of many of the cattle. And so in the parts of the Meerut, this work was carried out by the veterinarian in the Meerut as well as by my student Bhumika Chauhan to carry out the dissections and finding out the liver fluke. So this is how the dissection is done. The infected livers were collected from the domestic buffalo, which was slaughtered in the city abattoirs, and then transported to ice. Now, these adult flukes were collected, and the morphological studies of the light microscopy as well as the other studies were done. How they were done? They were fixed in ethanol for 24 hours, washed, then mounted, permanent mounts were made, and then they were observed under the very high powerful microscopes to understand, as well as even in Motic digital microscope and molecular genomic studies of DNA isolation, PCR, was later on done. In brief, I would like to just tell about the studies which were done, molecular studies. Individual flukes were recovered were washed repeatedly in phosphate buffered saline PBS. About 2 milligram of tissue was removed, and from each fluke tissue, sections were lysed with lysis buffer, whereby a fragment of the rDNA 5.S8S region was amplified from individual adult fluke lysates. And what was the primer that was used? This was the forward sequence. So I will use the black color to show you. This was the forward sequence and this was the reverse sequence, which and hence the PCR was carried out. Finally, the amplified PCR products were analyzed by electrophoresis. So what exactly, at which stage did we come in? By doing the phylogenetic analysis. That is, ITS gene sequence was used to carry out BLAST, that is basic local alignment search with the database of LCBI GenBank database. And the gene expression was analyzed using the computational softwares. And different motifs were found using the softwares called as Copia and Homer. And these motifs that we discovered for the first time as these motifs, and these functional motifs have been discovered by us is 6 functional motifs which carry out various functions. This is what the computational results are. Let me show you what are the functional motifs that we have discovered. The study identifies the presence of the 6 important motifs, and they are a role in protein folding and conformational stability, intracellular transport, antibiotic resistance and homeostatic processes, resistance to digestion by proteases, interaction with cell surfaces, extracellular matrix proteins and other ligands, sexual commitment, and inducing chemotaxis, DNA repair, cell cycle control, and so on. Okay, so this is all we have done, and this is the final that I end with, that is challenges on an integrated omics, and this was dealt in a lot of detail by Dr. Pratima yesterday. And this is a very good workshop that we have attended, because our future plan is to do proteomics of this parasite so that drugs can be designed against this parasite control. Thank you so much. Thank you.
Thank you, Dr. Monica.
Yeah. Am I still audible?
Yeah.
I'm visible? I'm
yes.
not even sure. Okay, I'm visible. Right.
Good morning, Dr. Monica.
Yeah, good morning.
I have 1 or 2 questions. So your title talks about gene expression analysis, but I believe you did not show anything about the gene expression analysis in this presentation. And the second is you also discovered some motif. What sequence did you use to find these motif? Were these DNA or protein, or what kind of sequences have you used?
The sequences, hello, am I audible?
Yes, you're audible.
Yeah, the sequences that were used were the methylation, and for the other sequences using these hello?
Yes.
Right. The sequences were for the functional motifs, that is the methylation, glycosylation. Should I show you again the slide so that I can come back to it?
No need to show the sequence. Which, no need to show the slide. Which gene sequence have you used to identify these motifs? And
the primer and the
there's no need to show the primer. I need to know the name of the gene that you use. Where are these motifs present?
The motifs are present in the lysed parasite that we had taken, and then we had run these to the PCR.
I'm asking which gene. These motifs must be present on some gene, right? So which is that gene that you have analyzed?
Can we have Dr. Monica?
Hello?
Yeah.
Yeah, I'm so sorry. I lost the connection. It was the rDNA 5.8S. Sorry, I'm very sorry.
So how
Hello?
come? Yes, I got you. So,
right.
So would rDNA 5.8S make a protein? Because you're talking of the motif function, which are all protein functions.
I think there is some problem in connection.
Okay.
So,
No.
yes, sir. We can go to the next presenter, I guess.
Yes.
So next presenter is Dr. Devyani Mule, and the topic of her abstract is fungal DNA methyl transferase, a key to invade to host world. Over to you, Dr. Devyani.
Warm greeting to everyone present here. Am I audible clearly?
Yes, you are clear, and your slides are also visible.
Okay, I'm Dr. Devyani Mule, Associate Professor, Department of Botany, Zakir Hussain, Delhi College. And my topic I choose for today's presentation is DNA methyl transferase in fungi. This is not my research topic. This is just the review I have taken for this oral presentation. So I start my presentation with my beautiful slide. That is, I feel the future is fungi. If we see the diversity of fungi and other organisms, fungal-like organism is amazing, with between 2.2 to 3.8 billion species predicted. If we see the percentage yet to be studied, that is again higher in the fungi, which is approximately 93%. And when we see the biomass, the global biomass, again the fungi is again at the top, and approximately 90% are terrestrial fungi and only 2% are water. When we compare the global mass and the abundance of organism, again you can see the fungi is at higher position. You all know the role of fungi, like they produce industrial enzyme, nutrient cycle, decomposers, biodegradation, mycoremediation. My PhD work was on the basis of on the mycoremediation, that some fungi they absorb heavy metals, and as well as model organism. Very recently, this month only, 10th May 2022, Lazlo Nag Laboratory has released a poster as they are using fungi as a model organism by keeping in the mind that phylogenetics with comparative and functional genomics and transcriptomics brings a new field, new site in the whole genome information available nowadays. When we see the soil composition, again, the 5% total soil composition, 5% is organic matter. Out of that, 10% are organism and 50% are fungi. This is my, research material which I use for the my PhD work, Ficus religiosa, and different varieties of ficus showing the fungal species on the PD agar medium. So life as we know it would quite simply be lost without fungi. As we all know, fungi has a role as a plant symbionts, key decomposers, soil carbon balance, mycoremediation, as I told you just in my previous slide, and food safety and human health, and moreover the pathogens. When we talk about pathogens, fungi can be a pain too. They can cause disease in humans, animals, and plants and destroy huge amount of agriculture produce. When we see the food security, that is with the unsafe food and food waste because of the crop pathogen, mycotoxin producer, infectious fungi, and foods spoilage fungi. On the other way, food security can be with the food as a fungi and the enzyme and total production of the food. Why fungi? Fungi can feed us, heal us, free us, or save our world. So I will mainly focus on the fungi because fungi and plants are the major stockholder. I will discuss about DNA methylation in brief and the pattern and function of DNA methylation in fungal plant pathogen. As we all know, I will skip this slide, that the DNA methylation is a methyl group added to the cytosine and adenine. And this methylation process activities change, DNA molecular activities change easily. And this methylation is it is a kind of 5-methyl cytosine. This is the presentation illustration of the methylation, which can lead to gene silences. And when we see the role of DNA methylation, that is importantly the gene regulation, silencing, and activation, and many more in the cell development and cell differentiation. This is again the illustration to show the methylated and non-methylated, hyper and hypermethylated in the gene silencing and as well as in activation. So this is again the demonstration, the presence of gene where the DNA methylation works. When it binds with promoter, it can stop the expression. When it binds when it joins at the transcription site, it stops transcription, and at the gene body level, this gene expression, elongation, and splicing takes place. If we talk about fungi, there are many roles in relation to the methylation process. So I will focus on pathogenicity and the secondary metabolites. This is again the same illustration in a different way. The reputation, defense mechanism, secondary metabolites, development, pathogenicity. When we see the process, DNA methyltransferase enzyme, which is a group of protein which is associated with fungal biological process. When we see, it is a very diverse, and it has taken attention of many researchers in this field, because in terms of this fungal aspects, the methyltransferase biological function, it has a role in the biological function, protein domain, and compared with the plants and animals. By seeing the comparison, this is the recent paper which has shows the comparison of mammals and fungi, and these are the the enzymes which are present in the process of methylation. So mainly methylation process in fungi is with the DNA methyltransferase, DIM, and DNMT1, DNMT5, and RAT. They are mainly present in the transposon region in comparison to promoter and gene body. Very few has been reported from the gene body. This is again the comparison of the plant, fungi, and animals, but I will mainly focus on the fungi. So when we see the ascomycetes and zygomycetes, mainly they were present on the transposon. Gene body methylation is totally absent in these fungi, and it was mainly in the DNMT1 and DIM2. As I earlier told you that DNA, so most of the fungi lack body methylation. So main focus is on the DNMT1, 5, and 3. These methylomes are connected with the fungi, and they have a role to present the taxonomy and the diverse uses of DNA methylation process in the fungi genome. Recently, 21, Nye et al., 40 fungal species has been studied for their genome sequencing, and DNA methylation profile has been studied, and it has been seen in Basidiomycotina. DNMT1 and DNMT5 has a role in the CG, and in Ascomycotina, the RAT and DIM2 has a major role. Again, it has been observed in the mycel, in the different stage of development of fungi, that low methylation level is in the mycelium in comparison to spore and other stages of the fungal growth. This can be seen in the pleurotus, that when the monokaryotic mycelium, which is the initial vegetative state, low methylation, and when it has been compared with the fruiting body, the dikaryotic mycelium, or I can say the tertiary mycelium, the slightly higher level has been reported. Same cases in other neurospora, that the vegetative stage as it's higher, and it is low in the sterile case. So many more information, but for that we need to study the genomic sequence of the fungi, and out of that, till date, 40 fungi has been reported for methylation, and only 14 for the fungal life state. I'm going to skip the other slides. It has a role in the pathogenicity, and it has been reported with the metrrhizium and the Galeria that 60% or spore germination is reduced, and this is very important for the pathogenicity. If the growth of the spore germination is reduced, it can lead to a more research area where we can work on this. If we talk about secondary metabolites, here again, the secondary metabolites production can be inhibited with the help of the presence of the you can say anti-methylation drug like 5-hexacetidine. So these are the role of the methylation. where we can work host pathogen interaction. So basically, when we will keep eye on this, so what we need the data, methylation data, the genomic data, epigenetic markers, so that we can go for future fungal research. So it has been, again, I told you, different percentage has been reported, and different groups of classes reported in the fungi. I believe that it's a key to invade the fungal host plant and yet to discover lot of things has to be discovered in this field. On a lighter note, even Mushmush and Michelle has given the, they have presented the Amazon series to protect the fungi. I also believe that we should, and I suggest we should watch these movies, or the documentary movies also. And very recently, Wild and IUCN has added the slogan that flora, fauna, and fungi we should protect. And thank you. I believe that fungi are the guard recycles of the planet and vanguard species of the habitat restoration. Thank you.
Thank you, Dr. Devyani. Over to the judges for questions.
Good afternoon, Dr. Devyani.
Afternoon, sir.
So my question is, if you look at plants and animals, DNA methylation essentially is in the cytosine at specific sequence residues. And now, since fungi is a different kingdom altogether, what kind of sequence residues are subjected to cytosine methylation?
They are CHS, CHSP. They are majorly studied in this manner.
Which one? CH?
CHH, CSG.
CHG, so not CG?
Yes, sir, CG also.
So in that respect, it is more similar to plants and plants rather than animals.
Yes, it is more similar to plants, but we have to work on this because all the DNA methylation has been not studied in fungi. Only few has been studied, and especially they are on the transposon, which is again in comparison to the plant and animals both. As I skipped one of the slide that it is CG and CH.
I have a different.
One is in mammal and another is both the type are present in fungi, both plants as well as animals.
And so do fungi also have small RNA mediated DNA methylation?
Yes, sir. Yes, sir.
Reports are already there, right?
Yes, reports are already there, but I have not mentioned because my presentation was my first presentation and it was already lengthy.
No problem.
So I skipped all those parts.
Thank you.
Thank you, sir.
Hello, Dr. Devyani. Just wanted to ask, is fungal DNA methylation any different compared to other organisms?
Yes, ma'am. As I have shown with that, the reference from the research paper also, that it is on the transposones as well as in the promoter region also. But when we see the comparison of gene body level, then it is lacking. This is where we can find more research work. We can try to find out in other fungus also, because as is seen in the aspergillus and pleurotus also. So more work need to be done in this field. But in comparison to animal and plants, they totally lack the gene body methylation.
Oh, I was precisely asking from functionality point of view.
I have not taken data on this aspect. Surely I will add in my presentation in future.
Okay, thank you.
Thank you, Dr. Devyani. So the next presenter with us is Dr. Sandeepa Singh. Yeah, hello, Dr. Sandeepa. Welcome.
Good afternoon, ma'am. I'm waiting for the present. Okay, yes, I'm presenting, ma'am.
Your slide is not visible yet.
Just a moment, ma'am.
Yeah, sure.
Just a moment, ma'am. Ma'am, is the presentation visible and I am audible?
Yes, you are audible and the presentation is also visible now.
A very good afternoon to all the organizers, the panel of judges, and the participants present here. I am Dr. Sandeepa Singh from Department of Botany, Day3 College, University of Delhi. And my title of presentation is Agrobacterium Mediated Transformation to Develop Citrus sinensis or Sweet Orange Transgenics. And I am the presenting author, and we both, myself and Dr. Misha Yadav have prepared the content. Just for an introduction, I have done my MPhil and PhD from Department of Genetics, South Campus, University of Delhi. So I'll begin with my presentation. We know that citrus is one of the most important nutritional and medicinal fruit crop in the world, and it is grown in more than 100 countries around the world. And it has high nutritional and cosmetic value also. And because it is grown in such diverse locations, it is susceptible to different kinds of pests and diseases also. And one of them is highly devastating and debilitating disease, citrus tristeza virus. Tristeza disease. Citrus tristeza disease caused by the tristeza virus. And this virus completely destroys the propagated crop that is propagated on lime, citrange, or grapefruit rootstocks. These rootstocks are highly susceptible to the tristeza virus. And to combat this, the genetic transformation is the only stable, reliable, and efficient alternative to combat the losses which every year run into more than $50 million around the world. So this is a highly devastating virus, and because of this only, most of the crop are destroyed around the world. And apart from destruction of entire crop, the fruit size, fruit quality, the juice quality is also highly reduced. And the plant virus is also the longest known RNA plant virus, and it has a 19.5 kb genome, and it is transmitted by the brown citrus aphid mostly. This is a phloem transmitted virus, and we can see the pictures how badly it affects the crop. So I would quickly rush into the experimental work. And up till now, the main transgenic strategies to impart viral resistance into sweet orange have been use of coat protein, replicase gene, movement protein, and RNA mediated resistance. Out of which coat protein mediated resistance has been found to be most stable and most effective, depending upon various reports published in the net. And I would like to mention that this work, though a little old, is the first ever report of successful sweet orange transformation in India, and also the first report of introduction of coat protein gene into sweet orange. Up till now, this is the first one, because this is a highly recalcitrant crop compared to other solanaceous and other dicotyledonous crops. This is citrus is highly recalcitrant crop which does not respond to agrobacterium mediated transformation. So we took epicotyl explants in citrus seedlings because they are quite responsive. And these were the main objectives of the study. First of all, devising a method for agrobacterium mediated transformation using Gus reporter gene constructs, and then a molecular analysis.
Slides are not moving.
Oh, sorry, ma'am. Slides are not moving, ma'am. Okay.
Yeah, now it's their objectives.
Okay.
Yeah.
Okay, ma'am. So I will keep it in this mode, not in full screen mode. Is that okay, ma'am? If I keep in this mode?
Yes, that's fine. Yeah, that's fine.
Fine, ma'am. So the objectives were to standardize the transformation procedure and do the molecular analysis for the gene that we had taken. We had taken the Gus gene for standardization of transformation, and then the same protocol was taken forward to develop coat protein mediated transgenics. And coat protein was taken from the virus growing in Kalimpong, West Bengal. This is the tDNA map of the standardization protocol, where it has NPT2 gene as selection marker and Gus gene as reporter gene. And this is the protocol, wherein the protocol is quite lengthy as compared to other crops. because it requires a long co-cultivation period also and a long regeneration time also. This crop is very, very slow in growing because it is a tree crop. Therefore, it is very, very slow in growing, and very small shoots are obtained in about 4.5 months time. So we had to do more than around 300 transformation experiments, independent transformation experiments, to get a few shoots later on. Some of them were, some of the experiments gave quite appreciable results, and some were quite bad. So this is the culmination of the standardization experiment, wherein we can see the putative transgenics in various stages of transformation from 1 centimeter long cut epicotyle explants placed on co-cultivation medium, and slowly we can see the regenerated shoots coming from the cut ends. And then moving on, in the last picture, we can see small shoots which were later transferred to potted plants, and the plants had grown to about 6-7 feet in height. The picture has not been included here. And this is the result of, ma'am, the slides are changing. Slides are visible?
Now they are visible.
Thank you, ma'am. So this is the result of GUS histochemical assay to ascertain the GUS gene integration in the transgenics. And here we have the stem section cuttings and the entire the shoots, where we can see that they have been uniformly stained by the X-gal. X-gluc, and the non-transgenics have not stained at all. They are completely pale in color, which shows that efficient transformation or efficient integration of transgene had taken place. And moving forward to PCR analysis, in the most successful set of experiments, we obtained between 5% to 23%. Okay, sorry, ma'am. So I would finally go to the final slide. This is the result of the transformation experiment obtained with the protein-mediated core protein gene, where we have the core protein transgenics or the real viral transgenics, which we want. And this is the stable integration of the core protein gene in the PCR. And this is the RT-PCR results of the transgenics now. And they will be further taken forward for further RNA analysis and virus challenge assays, for which we did not have time. Thank you, ma'am.
Thank you, Dr. Sandeepa. Now it's open to questions.
Hello, Dr. Sandeepa.
So good afternoon, Sandeep.
Yeah, go ahead.
I have one small question. So do you know the copy number, any kind of, have you done any analysis?
Yes, sir, we have done Southern with initial transformants with GUS gene. And we obtained a very hazy picture. That is why I didn't paste it here. And in one transgenic only we had a double copy number. Otherwise, all the other ones were single copy. I had done Southern of approximately 24 transgenics. One was testing negative, and one had 2 copy number, and one was the rest all were single copy.
Single copy. Okay.
And sir, Southern could not be done with viral transgenics, because once you integrate a viral gene, then the growth of the transgenic becomes approximately 1/5. So therefore, the transgenics were too small for a Southern analysis. Instead, we did Northern and RT-PCR and this thing.
But even with RT-PCR and this thing, you can still do a copy number. You know, if you have some sort of. So adjusted with the, you can do what you can do is you can convert this molar ratio of your spiking with the genome size of your citrus, you know.
Okay.
So you convert a molar thing, and then you can see if 1 copy gives me so much transcripts. So how much would 5 copy give me?
Okay, we can extrapolate in that manner.
It's inaccurate, but still gives you some idea. Anyway.
Okay, thank you, sir. I'll definitely do it.
Yeah, you can try. And the other thing is just a suggestion. You can There is one paper published about last month, I think, from NBRI, but by Dr. Debashish Chakravarti's group on why some plants are recalcitrant to being to agrobacterium transformation. So he's done a sort of study. Okay, just look at that.
So that is why only we had to do more than 250 transformation experiments, which were recorded, and the unrecorded.
If you read that paper, maybe it's to do with give you some clues.
Yes, sir, definitely, definitely. I'd search it right
Thank
away. Thank you so much, sir.
Yeah, Dr. Sandeepa, just a small query. Are there any other methods to develop transgenics in citrus?
Ma'am, I'm sorry to say that this is the although agrobacterium is highly reliable and a very efficient method to develop, but it also fails many of the times. Therefore, we have to undertake so many experiments. And beyond this, no, ma'am, nobody has developed transgenics in citrus, especially in sweet orange, apart from this method.
So why did you try any other method?
Sorry, ma'am?
I'm asking, why did you not try any other method?
Ma'am, because by this method only, we took almost 2.5 years to develop transgenics. So trying and trying other methods, I think would have taken too much time without any appreciable result. So that is why. And at that time, only this method was found reliable. And even up till now, I haven't come across any report which says that other method can be done. Although we can take different explants, but the method is agrobacterium, through agrobacterium only.
There are methods available, biolistic methods of transformation. So I think, you can just review and methods, right?
Yes, ma'am. We can.
Another, just a query that I have is, have you not published this work before?
Yes, ma'am. I had published only a regeneration thing. My regeneration paper is there. And for transformation, maybe I am unlucky that I have still not compiled the entire report. This is going to be an extensive report. I published other papers, but this one is still waiting.
Okay, best
I'll do
wishes.
it right away.
Okay, thank you.
Thank you, ma'am. Thanks very much, all of you.
Thank you, Dr. Sandeepa. Now, the next we have Harneet Pal Kaur with Saroj Arora, and the title is Modulatory Effect of 4-methyl thiobutyl isothiocyanate isolated from Erica sativa in combination with paclitaxel for the treatment of breast cancer cells. Over to you.
Hello, yes, good afternoon, ma'am. Am I audible?
Yes, you are audible and your slides are also there.
Okay, thank you. Good afternoon, everybody. Today, I have Neerpal Kaur, Research Fellow of Professor Saroj Arora at the Department of Botanical and Environmental Sciences from GNDU, Amritsar. Today, I'm going to present the modulatory effects of 4-methyl thiobutyl isothiocyanate isolated from the Yucca sativa in combination with paclitaxel for the treatment of breast cancer cells. So let's begin with the introduction. Hello. Am I audible, ma'am?
Yeah, you are audible. Please go ahead.
You are audible.
Yes. So coming to the introduction, cancer, as we all know, it is a multifactorial disease characterized by unregulated growth, dysregulation of cell signaling pathways, and dissemination of malignantly transformed neoplastic cells. Breast cancer is the most common cause of death among women in the whole world. There are numerous treatment options like chemotherapy, radiation therapy, surgery, but these conventional chemotherapy drugs are effective, but still they have severe side effects. So to overcome the side effects of conventional drugs, natural chemotherapy may works better. I skipped this slide. So come to the next. So current studies. So currently we have focused on the use of isothiocyanate, particularly the 4-methyl thiobutyl isothiocyanate as an anti-cancer compound to lower the toxicity of conventional chemotherapeutic drug with the combination of paclitaxel against the breast cancer cells. So firstly, introduce the Rucus plant. It is an annual herbaceous plant distributed in the Mediterranean region. Its common names are rocket salad, Rucola taramira. Its seeds are rich source of glucosinolates that are break down into isothiocyanates. So extraction of 4-methyl thiobutyl isothiocyanate was done by preparing the powder of Ficus tiva seeds. Then the extraction of oil was done by using Clevenger apparatus. Then the following procedure has been performed. So then the extraction of 4-MT-BTC was done by using thin layer chromatography, in which the single peak was observed at 26-27 fractions of crude extract. Then the quantitative analysis of 4-MT-BTC was done by using GHPLC, which showed a single peak at a retention time of 13.1 second. Then the chemical structure was further confirmed by NMR analysis, and then we performed genoprotective studies of 4-MT-BTC, in which we performed plasmid DNA nicking assay. What we found in plasmid DNA nicking assay, we have used 3 concentrations of 4-MT-BTC that are 25, 50, and 100 micromolar. And what we observed, 4-MT-BTC showed maximum DNA protective activity at its highest concentration of 100 micromolar. Then we performed anti-proliferative studies by using MTTSA, in which T47D breast cancer cells have used, and we have performed 3 groups. First one is the 4-MT-BTC treated T47 breast cancer cells. Then we give treatment paclitaxel. Then we give treatment with the combination of 4-MT-BTC and PAX. And we found that at the different concentrations of 4-MT-BTC were used, 2, 5, 10, 20, 40 micromolar, and same way paclitaxel have been used as a 0.1, 0.2, 0.4, 0.8, and 1 micromolar. Then after knowing the IC50 of these two natural compound and conventional chemotherapeutic drugs, then we proceed to take it as a combination, and by using a Chow and Tale method, we have found the combination in 1:1. That is the best ratio at which they both show the synergistic anti-cancer effect. Then intercellular ROS detection assay was performed by using 2,7-dichlorofluorescein diacetate staining, in which we found that the maximum accumulation of intercellular ROS was detected in the cells which are treated by combination. And then we proceed to the measurement of mitochondrial membrane potential, in which we have used rhodamin 1,2,3 dye to treat the breast cancers, untreated breast cancer cells and the treated breast cancer cells. And it showed that 4-MT-BTC has a 27.9% of intact cells population, while the paclitaxel at 0.1 micromolar, it showed 23% of intact cell population. But in the combination, the intact cell population decreased by 17.7%. So we can say that the combination has best activity at the 1:1. Then we proceed to the Western blotting. In the Western blotting, we have studied caspase 9, caspase 3, P53, and BCL-XL protein in the T47D cells, in which what we found, caspase 9 and caspase 3 activities were significantly increased in the combination treated cells, while P53 also increased, but the anti-apoptotic protein, that is BCL-XL, was decreased. So by studying these proteins, we can conclude that the apoptosis has been done in the T47D cells by the 4-MT-BTC and PAX and the combination. Then we proceed to RT-PCR analysis, in which we have analysis of 4 genes, BCL2, cytochrome C, and P53 and APAF1. Then we have studied 4-MT-BTC packs and their combination in combination treated T4070 cells, in which what we found that the P53 expression was gene expression was highly upregulated in the combination treated group. So by this way, we can say that the 4-MT-BTC has enhanced the paclitaxel cytotoxicity. Thank you, ma'am.
Thank you. Open to questions.
So Harmanpreet, I have one small question. So how much benefit do you get out of this isomethyl cyanide over paclitaxel?
Sir, 4-MT-BTC has decreased the paclitaxel dose by 4%.
So is it statistically significant?
Yes, sir. Sir, firstly, we have studied their IC50s. Then in combination, what we found that their IC50 was lowered by the 4%. So according to previous research, what we मतलब जैसे हमने वहाँ पे भी देखा तो synergistic anti-cancer effect था.
Thank you.
Thank you, sir.
So now, next, thank you, Harneetpal.
Just one small question.
Yeah, one question from
Dr. Harneetpal. Just wanted to know, any other member of Brassicaceae has this 4-methyl thiobutyl isocyanate? My particular species.
Brassicaceae family, ma'am.
Yeah.
Okay, thank you.
Eureka stiva from the plant of Eureka stiva.
Have you done any in silico studies to see the molecular interaction? Oh.
No, ma'am. Not yet.
Okay, thank you.
Thank you, ma'am.
Thank you, Dr. Harneetpal. Now we are going to we are left with only 2 presentations now. So the next presentation is next presenter is Bishun Dev, and the Bishun Dev Prasad.
Yeah, hello, ma'am.
Yeah, hello. So, and the topic is Illumina-based de novo transcriptome sequencing in safflower. Welcome, Dr. Bishun Dev.
Thank you, ma'am. I'm trying to upload my slides.
So if you are going to take time, can we move on to the next?
No. It's there.
Okay,
yeah.
Fine.
I hope you can visualize my slides.
Yeah, it is there. The slides are there.
Good afternoon to all. The topic of my presentation is Illumina-based de novo transcriptome sequencing in safflower. I am Dr. Bishundev Prasad, working as an associate professor at Dr. Rajendra Prasad Central Agriculture University, and Dr. Sangeeta Sahni is my co-author. She is an assistant professor in the same university. So today I'm going to talk about safflower and why we say that climate resilient crop. Then we will switch to glucosinate. Then we'll see the effect of glucosinate on stress tolerance and yield. We'll see also the effect of glucosinate in safflower. Then we'll go to de novo transcriptome sequencing, and we'll also try to identify the glucosinate biosynthetic pathway in the sequencing data that we have obtained. So here I'm trying to show that this climate change, we cannot stop it. It has to come. So what we can do is try to mitigate the effect of climate change. So safflower, since it is, although it is a it's a oilseed crop, but this has a potential to mitigate this climate change as it's a drought tolerant crop. and it can be promoted in the dryland ecosystem. So it will be a suitable crop for the climate resilient agriculture. Brassenestride, as you know, that it has the various role. It's a plant steroidal hormone, and it is known for its plant growth and development, as well as it is also known to protect the plants from variety of experimental stresses. And these are my previous work, where we have also published some of this in scientific report in 2016, where we have shown that if we apply the brassenestride in brassica and brassica nippers, you can see it will enhance the thermotolerance. It also has the capacity to mitigate the drought stress, since its root got elongated, and it also leads to the enhanced yield. We have observed 10 to 20% increase in the seed yield. So keeping this in mind, we thought whether this EBR or brassenestride has some effect on the safflower or not. So we provided the exogenous brassenestride to the safflower seeds, and in the seedling we observed there is a huge increase in the thermostress tolerance. So then we move ahead and we try to identify what are the different brassenestride pathways or brassenestride genes are there, because none of the genes or brassenestride pathway has not been reported yet. So we took we took the treatment, brassenestride treatment, that is EBR, and MOT treatment, and we extracted the RNA and we did the Illumina sequencing, and this is the workflow that we have used. We did the raw reads, then we filter and we got the high quality reads, and then we go ahead for the transcriptome assembly, and then we predicted the coding sequences. We also did different analysis that I'm going to show in the subsequent slides. These are the summary reports that we have obtained is approximately 5 GB in each sample, so total 10 GB data was obtained, and these are the different criteria that we have followed, and this N50 was found to be approximately 1.5 KB. And this is the distribution of transcript. We got the maximum transcript in range between 200 to 500, but we also got quite a good number of transcript that is ranging from 1,000 to 2,000 transcripts in case of transcript, as well as the predicted genes. Also, we got the very good amount of transcripts that is ranging from 1,000 to 2,000 base pair, that is 2 KB. So we can say that the data that we have obtained is of good quality. Then we identify the differentially expressed genes. These are the heat maps showing the differentially expressed gene in control as well as in EBR. Then we also identify some of the transcription factors, because these transcription factors play a very important role in case of stress condition. Interestingly, you can see that we have also obtained this best one, and this best one is known as the brassenestride regulated transcription factor. So some of the genes also has this best one and other stress related transcription factors, which says that this brassenestride may play a major role in increasing the stress tolerance in the safflower. Then we also performed this QRT-PCR analysis to see whether the data that we have obtained is matching with the QRT-PCR analysis or not. We have randomly selected the genes and we have checked the expression value using this real-time RT-PCR analysis. And this is the TED pathway analysis, and you can see here in the signal transaction pathway we have obtained a huge number of genes. So whenever we apply the brassenestride, some of the genes will get activated in the signal transaction pathway, and we have obtained more. Some of the genes are also upregulated in EBR condition. Now this is the MapMan analysis we have done, and here you can see we have got 6 genes which is known for brassenestride. So brassenestride pathway genes has been identified, and we have identified 6 gene in the safflower when we apply the brassenestride treatment. And this is the TED pathway, which is showing that we have obtained this DWAF4. DWAF4 is the rate limiting step in the brassenestride biosynthetic pathway, and in our previous report in brassica we have also showed that when we overexpress this DWAF4 genes, then we have obtained the increase in the thermotolerance, as well as we have got the increase in the drought stress, and we also shown the enhancement of the seed yield. So if we apart from this drop, we have obtained other genes. So then we wanted to see where this dwarf is localizing. So for that purpose, we have done the confocal analysis. We have cloned this. First, we have fused this dwarf with the BiFP, and then we infiltrate in the Nicotiana benthamiana leaf, and this is the confocal studies. It is clearly showing that it is going to the in the membrane, transmembrane, as well as in cytoplasm. Then we also move ahead and we clone this in the tDNA vectors, and we try to overexpress in the sap flower. You can see here, this is the way we have used genetic transformation in the sap flower, and this was confirmed using the PCR analysis, and the expression value of the dwarf genes was also analyzed using the QRT-PCR analysis. So this is what we have done. Okay, so I'm coming to the last slide. The part of some of the work that we have shown here has already been published in Journal of Plant Growth Regulation, and some part is remain. We are trying to finish this. Thank you very much.
Good afternoon, Dr. Prasad.
Hello, sir. Good afternoon, sir.
Good afternoon. So what is the genome size of safflower, and how much is the, I mean, what percentage of the transcripts are sort of found, I mean, you detected in, I mean, in the samples that you sequenced? I believe it is a
transcriptome sequencing, sir. It's not a genome sequence. It's
I know.
Sequence.
I'm saying, what is the genome size and the number of genes known in safflower? If it is known, and what percentage of that is represented in your data set?
Sir,
Your screen is frozen. It has freezed, Dr. Dev. Hello, is it audible?
Yeah, you are audible, but yeah, he's here, Vishal.
You are on mute. Kindly unmute yourself.
So nearly we have obtained 8,000 genes transcript we can able to report here, and I hope the transcript, the whole gene transcript size may reach up to 20,000. So approximately 50% of transcript we can able to find it out.
Some more questions from the judges?
I think Professor Das has already asked this question, but since he's not there, I would just like to ask, in your abstract, you have mentioned that most of the hits you obtained were with Cyanara. Why not with
Yes. This is also very close to the safflower family. It's also an oilseed crop, and but not used commercially for extraction of the oil.
But there are reports, you go through the works of Wu et al., which is about Carthamus itself. And
sure, I will go through.
Okay, thank you.
Thank you very much.
Thank you, Dr. Dev. And now we have Ananya Bandopadhyay. No, I'm so sorry. It's B. Hariram Das. And the topic is assessing the effects of anthropogenic activities on river microbiome, a multi-omics approach. So can we have Dr. Hariram Das? Dr. Bishnu, Dr. Deo,
Yes.
sorry, you may stop presenting your slide.
Okay, sorry, yeah. Yes,
Thank
yes,
you.
I'm
Thank
doing
you.
that.
Ananya is the presenting doctor.
Yeah, yes. Ananya is present. Okay, sorry.
Yeah.
So it is Ananya Bandopadhyay.
Yes, ma'am. Good afternoon. Is the screen visible?
Good afternoon. Yeah, it's visible.
All right. So good afternoon, all. I am Dr. Ananya Bandopadhyay. I'm an assistant professor at Department of Zoology, University of Delhi, and along with my senior colleagues, Dr. Ilmas Naqvi and Dr. B. Hariram Das from Zoology Department, Zakir Hussain Delhi College, University of Delhi. I am going to present on our behalf a review of the latest studies on the effect of anthropogenic activities on river microbiome and how this can be addressed by a multi-omics approach. So first things first, so much of omics that we have learned in this workshop. I'm going to talk about meta-omics in my talk in this presentation. So when we talk about river microbiome, basically it is a collection of all the microbiomes that a collection of all the microorganisms that are present in a given defined environment. So when we talk about river microbiome, it is the collection of all the microorganisms present in a given sample of river water. Now, but the microbiome, because it has a collection of many microorganisms, they cannot be studied directly, and therefore it must be, because it cannot be cultured, all the microbes cannot be cultured in the laboratory conditions. Therefore, comes into picture the concept of metagenomics. Metagenomics is basically all the genomes of the microbes that are present in a given environment. So what we are going to get through metagenomics or the metagenome is the microbial potential, that is in a given sample what all microbes are present, what are the cellular material that is available in a given river sample. Apart from that, when we study the genomics of these microbes, we also get an idea about what is their potential, which is known as the metabolic potential, what they can do for us. Whether they are doing it currently, presently, in a given point of time, that we are not sure, but at least in their genome, they have those genes which have the potential to do some specialized function. So that would be, so all these are being currently studied as part of metagenomics. However, to understand the full potential and to understand how these microbes are behaving in a given water sample, whether it's a pristine water, whether it is the headwaters from where the river originates, whether it's almost towards the mouth where it becomes a continuous part of an ocean or a lake where it ends, or whether the river is in a very polluted, heavily polluted condition, that would change the kind of genes expressed in the set of microbes that are present in any river water. And there comes the third part of this, which is what kind of functions these microbes are doing at a given point of time. And this is where three more omics concepts come into picture. But rather than omics, these are more like meta-omics. So the concept of meta-transcriptomics that I have mentioned here. So meta-transcriptomics is the analysis of all the gene transcripts that are present, which can be sequenced by in a high throughput manner, and give us an idea about the total set of RNAs that are present in a given microbiome. Along with that, we have metaproteomics, which tells us that what all expression mRNAs are being converted into proteins, which means that those proteins are actually carrying out those functions. And along with that comes the concept of metabolomics, which is also known as metabonomics. The term has been coined such, which means that maybe we can look into novel pathways that these microbes have evolved so that they can handle the kind of pollutants or maybe the different kinds of situations in which they have to thrive in these river waters. So with this overview of what we are looking at when we talk about river microbes,
slides are not moving.
Yeah, so I'll move to the next slide now.
Okay.
Has it changed?
Yes.
Yes.
All
yes.
right. So with this overview, I'm starting with some of the studies. So this is a review of the recent studies that have been done. And therefore, this table enumerates the major rivers around the world where, the microbiomes have been studied. For example, the Amazon River, the Danube River in Europe, Nile in Egypt. Even in India, there have been studies on Yamuna and Ganga, Narmada, Kaveri, and Gomti. So here we have tried to show the dominant microbial communities that are found. So there could be across the various rivers, you see that the microbial communities are so different from each other. There could be, in some cases, there is a dominance of proteobacteria. There could be a dominance of, actinobacteria. And when we, look at different rivers, the microbiome of different rivers are different. Not just they are different in different rivers, what we have also seen is that even in a single river, there can be temporal variation. So there's a very interesting study done by an Indian group on the Yamuna River water that was collected from the ITO site of Yamuna. And they have found that the microbiome changes between the month of June and the month of November. So they had taken two samples, month of June and month of November, and they saw and they analyzed the microbiome, and they found that there was a drastic change between these two samples, pre-monsoon and post-monsoon. That is what they have tried to show. So basically what I want to So here we see that there is temporal variation. Similarly, there are spatial variations where we see that from different locations in the same river also we can have these variations. Now we come to the pollutants. So pollutants basically come from the sewage or garbage to the landfills, from the wastewater treatment plants into the drinking water, and through the drinking water, it again reaches into the organism. So this is a major issue. And along with the pollutants, it's also antimicrobial resistance. So it has been found in rivers like in Canada, Fels River, Bogota River in Colombia, in China, in Yamuna River in Delhi. There are many such antimicrobial resistance genes that have come up, which provide resistance to broad spectrum antibiotics, very common antibiotics that we use in our for medication purposes. But that is all very bleak situation, but nonetheless we still have potential and a silver lining to this entire thing, where bioremediation potentials have come out from the microbes that are present in these polluted rivers. And it shows that how multiple such species, genus, they are available, which have the potential or have the genes by which we can carry out bioremediation, biodegrade xenobiotics. So in the end, I would just quickly summarize that using these multi-omic approaches, we can, not just see the microbial diversity, not just see the variation, spatial and temporal variation across the river, across time, but at the same time, we can get, putative gene candidates which can have bioremediation or xenobiotic degradation potentials, not just selective genes, but also at the same time we can maybe identify entire pathways to degrade major industrial pollutants, for example, toluene, heavy metals, hydrocarbons, organic waste, and at the same time, the antibiotic resistance genes that are coming up that must be taken care of at the wastewater treatment plants before they can be discharged into the river water, so that we do not have more of these antibiotic resistant strains coming up. So using this integrated meta-omics approach, we would be able to revive the river microbiome, and that is essential for keeping it as these are very crucial for the biogeochemical recycling of river as an ecosystem. Yeah, I'm done. I'm really sorry for exceeding the time. I'm done.
So Dr. Ananya, good afternoon.
Good afternoon, sir.
So my question is very simple. Would an approach be universal for studying rivers that are perennial in nature and rain-fed in nature, as you find in India?
No, sir. Of course, as we have seen in case of Yamuna, that there is a temporal difference, And similarly, microbiome being an ever-changing, so dynamics of microbiome are very different.
So that means how would you sort of come to a conclusion, because it's going to keep changing?
Yeah, yes, sir, very much I understand. So I do not have a ready answer, or this is a huge question, because microbiome is something that we have just started to explore in the last 15 and 20 years. So once we get the data, we get a lot of data from all across the world, maybe from India also, we can then start thinking about bioremediation approaches, or how these can be revived and the microbiome can be restored and kept in a natural form. This is a huge area that has just started to show its potential. So answers right now, trying to get an answer right now will be difficult, but I think as soon as we start to restore the microbiomes in their most natural form, as long as we are able to keep the pollutants, the xenobiotics away from the rivers, I think the microbes have the ability to revive themselves if as and when they are given time. So I think that could be a starting point.
Yeah, thank you.
Hello, Ananya.
Hello, ma'am.
Yeah, I just wanted to ask, are there any success stories, whether in India or abroad, using this technique? Such studies.
So what we have seen so far from the papers that we have studied, that scientists have just started to look into the transcriptomics and metaproteomics. The basic work that is still being done is in the field of metagenomics. They are trying to identify the putative genes at the metagenomic level, but they have not looked into the metatrans. There are very initial studies of metaproteomics because it's difficult to do, and so these are only explorative right now, but based on this, I have not come across a study where they have tried to restore the microbiome of a river so far.
Thank you.
But then, ma'am, these can be taken up from there, and then in the wastewater treatment plants, these microbes can be introduced so that the wastewaters are treated first before they are discharged into the river. That can be done.
So definitely, this is need of the hour, and if this really works, probably it will create wonders.
I completely agree.
Thank you.
Thank you so much, ma'am.
Dr. Tabassum, unmute yourself.
I'm so sorry. So
there
I
is
guess
one
yes, ma'am. But can we take it afterwards, because we have head Department of Botany with us for her address, for her valedictory address, but she has to join at 1 to some other meeting. So is the presenter here? Archana Raj is there?
Yes, ma'am.
Okay, so we can take it after the address of head. Is it okay, ma'am? Okay.
Okay, ma'am. Okay.
Is it okay, ma'am and
it's
Sandeep?
okay with me, but what about Professor Das?
Yes.
Yeah, fine with me.
Thank you so much, sir. Now I would hand over to Dr. Vatal to introduce our head Department of Botany and for the further proceedings.
Thank you, Dr. Tabassum. With this, we come to the last segment of 3-day event. I have the honor of introducing Professor Suman Lakhanpal, who's presently the head Department of Botany, University of Delhi. I have known her since she joined the department for her for her MSc, and I also take pride in the fact that I had the privilege of taking a few biochemistry classes of her batch when Professor Sacha was abroad and I was a PhD scholar. Coming to her formal introduction, Professor Suman Lakhanpal is Senior Professor and Head at Department of Botany, University of Delhi. Professor Lakhanpal did her graduation, post-graduation, and MPhil from University of Delhi. She obtained her PhD degree in botany in year 1989 from University of Delhi. Her research interests include plant molecular genetics, crop genetics, molecular analysis of biological diversity, multi-partner interactions, She has guided several PhD and MPhil students with more than 30 publications to her credit. Professor Lakhanpal is a member of editorial board of several international journals. She has edited several books and contributed several book chapters. Professor Lakhanpal was awarded the National Science Talent Search Scholar Fellowship and availed the fellowship for the entire education period, that is up to PhD. She secured second position at all India level in Agricultural Research Services examination 1985 batch held by UPSC. In the and her specialization at that time was genetics and cytogenetics. Professor Suman was also adjudged as excellent teacher and nominated for the award of best teacher in IARI. With this brief introduction, I invite Professor Lakhanpal to address the participants. Over to you.
Thank you, Professor Ratnam Kaul. Am I audible?
Yes, yes, you are.
Thanks a lot. And I'm sorry for the slight change in the program. Because of me, I have a commitment at 1 o'clock. I thought it will be over by 12:20 or 30. I can understand. I think there is a very enthusiastic participation by the presenters. So, respected Professor Narendra Singh, Principal of Sakristan College, if he's there, and distinguished resource persons and judges, members of my botany fraternity from the college, Dr. Ratnam Kaul, Dr. Malti, Dr. Babita, Dr. Tabassum, Dr. Devyani, Dr. Ruchi, Dr. Wahid Ansari, Dr. Zishan, Dr. Savinda. If I missed some names, I'm sorry. Members of the organizing committee and dear participants. I'm extremely happy to know that this 3-day event on International Conference cum workshop on Proteomics, Transcriptome and Molecular Drug Design and Integrated Omics approach is going to be completed soon, very successfully. I'll try to be more brief than what I had thought. The importance of each one of the 3 terms which you have taken in your title, they cannot be overemphasized. In the simplest expression, it reminds you of, DNA, central dogma of molecular biology. DNA makes RNA makes protein, or what we say, the mool mantra of the entire, life on this planet. So as you know, scientists have been calling this epoch as the Anthropocene epoch, though not officially accepted, but unofficially it has been called as the Anthropocene epoch. Similarly, this era has been called as the era of genomics. In fact, of late, people have started calling it as post-genomic era, or even, omics era for that matter. So it was a very right selection of the topic which you chose for this conference and workshop. I'll just briefly, for the sake of students, because we are teachers and a teacher remains a teacher all his life till he dies. So very briefly, I will just highlight some points that every genome, all genome of the living organisms has a coding part and a non-coding part. The non-coding part has been variously called as the dark matter, or even selfish DNA, not selfish gene of Dawkins. Doolittle and Sapiens has called it selfish DNA or parasitic DNA. So there's a lot which has to be studied about that part of the dark matter of the genome, what we call. But there is a coding part of the DNA genome fraction, whether it gets translated into proteins or not. The transcribed part of the genome, fortunately, is amenable to investigation, and that is what we are trying to study in the transcriptome. And fortunately, whatever we know about life on the planet Earth is coming from our understanding of that part of the genome. We know a lot, but the techniques and methods to know more about it are changing on a daily basis. So you could understand initially one or few genes at one point of time, and now with the recent advancements in the techniques, we can study, in totality, that is the spirit of the omics, a lot can be studied in one go. I'm getting a call from ex meeting. Okay. So, one thing where I want students, this is only for the sake of students to remember that unlike genome, when we say whole genome, we have sequenced every bit of the genome. We can say that we know about the whole genome, but when we have a transcriptome data, it is only a snapshot. It is only at one point of time in the stage of the life or in the time of the organism we are trying to study. It's like, in a very simple way, it's a snapshot. If we go to a place, if we go to Shimla, we take one picture, that is not Shimla. There are things beyond that, there are things after that, there are things left and right. So transcriptome and proteome are both very dynamic. This is one thing students should definitely get the message. In very simple words, I thought I'll explain more. I'm sure all of the very distinguished faculty members or resource persons must have already explained, but I thought I can't resist saying that from one genome, there can be many transcriptomes, and from one transcriptome, there can be many proteomes. So this is a very dynamic thing, unlike genome. So transcriptome and proteomes are more dynamic, so more interesting to study than the simple genome, what we call structural genomics. This is now functional genomics, so it makes it more challenging. Very briefly, I'll come to the third aspect of your topic, which is the drug design. And again, I would say post-pandemic, I think nothing can be more relevant than this, that there's a constant battle going on between the pathogens and the drugs. We have to do research to develop newer and newer drugs. Pathogens don't have to do research. They have to just mutate. It is their intrinsic ability to throw out with more strains, different types of strains, less virulent, more virulent. But every time nature throws a a different strain at us, we have to carry out a lot of research on meeting that challenge. So the newer tools are required every time we have to come out with newer drugs. So I would say a trained manpower in the three key areas which you have chosen is the demand of the day. To sensitize people to these three very important key areas is really, is the most relevant thing to do, and nothing can be more relevant than this. So it's a matter of great satisfaction for me that you have chosen such relevant and such appropriate topics for this workshop and conference. On a very personal note, I would say that when few months ago, my young colleagues from Zagreb College had first time come up with the idea of holding a workshop and a conference, I was a bit weary, I was a bit nervous, I should say a bit worried, because the worry was similar to when your child or a younger sibling does something more adventurous. You really want them to succeed, but you have a prayer on your lips that nothing should go wrong. And it's a matter of such a pride and satisfaction for me that the way they organized, they conceived, they executed, they delivered the first workshop, first event that the college, these people had done, was a pleasant surprise to me. And that led me to immediately say to Dr. Ruchi and Dr. Tabassum that this should be the beginning. You have to organize many more such events. And I'm so happy that they have proved themselves. They are going, by leaps. One after other, they are organizing such things. And I would request if Dr. Professor Singh is there, to please take a note of it, that you have a very energetic, very dynamic, highly, capable team of young college teachers who are capable of putting your college on the map of India for training the young students. Please definitely take a note of it, provide them with all facilities, all encouragement, so that not only within the country, they should be able to put your college on the map of the world by becoming a center of excellence for training the young scientists. young junior colleagues, for sensitizing them to the most relevant topics. The students, they are the right people, the link between the young students and the, scientists who are lost in their labs. So with these words, I would like to once again put my highest appreciation on record for the organizers of the team. I'm grateful to the college authorities for giving this kind of environment, for this, particularly to the senior teachers of the botany department, for providing my young colleagues such a positive, such a motivating environment that they are able to execute what they want. I understand, I know many stories from different colleges. It is exceptional the kind of support my colleagues are getting from the senior teachers of the college. And so I would convey my best wishes finally to the participants, and I really wish them all success to all the endeavors which they will be building on the foundation which has been laid in these 3 days. I wish them good luck, and I wish them to remember the resource persons and the organizers and the teachers who gave them this opportunity. So with these words, I thank you all, and particularly for making me a part of it. I wish I could spend more time on it. Thank you. Thank you so much.
Thank you, Professor Lakhanpal. It was really very nice of you to have literally stolen minutes from your meeting. I was much disappointed when you texted saying that you have a meeting at 1 o'clock, because it was this conference at the international stature was not possible without your support and without your guidance. So in fact, the organizing team at Zakir Hussain Delhi College is grateful to you for providing so much of support and having confidence in the college team to literally, it was a very high flown topic that they could do it. And it was definitely Ruchi's baby that she had been, imagining for the last 1 year that she needs to do it. So thank you once again. I won't hold you for long and look forward to another opportunity to hear you out. Thank you.
Thank you. Thanks so much. I'll be logging off. Thank you.
Yeah, thank you. Over to you, Dr. Tabas.
Yeah, ma'am. Thank you. So we can resume the oral presentation session. There's just one presentation that is left. It's Archana Raj. Can we have you here?
Ma'am. Yes, ma'am. Hello, am I audible?
Yeah, you are audible. So can you please present? Yeah, the title of the abstract is In Silico Molecular Docking and Pharmacokinetics of Selected Phytochemicals Against BK Channels.
Thank you, ma'am, for the introduction. Esteemed dignitaries on the dais and my dear colleagues, a warm good morning to one and all. I, Dr. Archana Raj, MBSc Scholar, Department of Veterinary Pharmacology and Toxicology, SIVAS Thrissur. I am here today to have a discussion on my research in silico molecular docking and pharmacokinetic study of selected phytochemicals against BK channels. My presentation is structured as shown here. The plant given here, Artemisia annua, reminds us about a major breakthrough discovery in the field of phytopharmacology, which led to the development of an anti-malarial drug, artemisinin. Likewise, thousands of known and unknown phytochemicals are present in the worldwide, but according to the work conducted by Saxena and co-workers, the major class of phytochemical present in the plants is phenolic group, followed by terpenoids and steroids, and later alkaloids. Even though there are innumerable phytochemicals present in the plants, the pharmaceutical industry not yet succeeded in manufacturing and marketing all these active entities into the market because of the cumbersome procedure. But with the advancement of modern technology, the development of computational models and simulation techniques lead to the rapid expansion of the global drug discovery process through high throughput screening procedures. In this study, we adopted the in silico tools to assess the activity of the flavonoids and terpenoids on BK channels. BK channels are big conductance calcium activated potassium channel, which is expressed in several tissues and have a major role in smooth muscle contractility, neuronal excitability, neurotransmitter release, and endocrine secretion. Consequently, the pharmacological modulation of BK channels, either their activation or inhibition, would present a plethora of therapeutic opportunities such as cardioprotective effect. On the ground of these facts, we planned the study with the given objectives. The receptor structure were downloaded from the RCSB protein data bank with PDB ID 3U6N, and the ligand structures were downloaded from PubChem. Both of them were modified for docking in MarvinView software. The docking procedure between the BK ligands and BK channel and the various ligands were carried out in Autodock 4 with a specified grid dimension. And the post-docking analysis and visualization of results were done in two softwares, Discovery Studio Visualizer as well as LickPlot Plus. After docking, the selected ligands were assessed for the pharmacokinetic parameter in the ADMETSR open source software. The given graph shows the binding energy of different ligands with BK channels, where the blue bar indicates the terpenoid class, gray bar indicates the flavonoid class, and the red bar indicates the standard compound andolase, which is a BK channel activator. From this graph itself, it is very evident that the gray bars or the flavonoid class have better docking scores than the terpenoid family. Among the flavonoid family, there are three members, myrcetin, camphorol, and quercetin, which showed good docking scores comparable to that of standard compound andolase. The post-docking results were analyzed in Discovery Studio Visualizer for the location of the interaction site. And the hydrogen bond interactions and the bonding distance of these three compounds were obtained from the LickPlot Plus. From the study, it is revealed that the myrcetin and andolase form a hydrogen bond at the similar site. And later, these three selected ligands, myrcetin, quercetin, and camphorol, were processed in the ADMETSR open source software for the prediction of the pharmacokinetic parameters. From the results revealed that the myrcetin and quercetin have a good pharmacokinetic profile and which can be used as the drug candidates. From the altogether, we concluded that the flavonoid class have better activity on BK channel than the terpenoid family, whereas the myrcetin and quercetin can be used as potent drug candidates for BK channel, and it can also be taken as lead molecule for further in vitro and in vivo assessment. Also, future studies should be carried out on other flavonoids for the assessment of the activity. These are my references. Thank you.
So good afternoon, Dr. Archana.
Good afternoon, sir.
So this is your PhD work, you said?
No, sir. No, sir. I am a PG scholar, MBSc scholar, veterinary science.
PG. Okay, so this is your PG research.
No, sir. Yeah, part of that, sir.
Part of it. Okay, and so you talked of some plant. I've missed the first part. The one you said something about the Artemisia is taken from a review article, you said, because that's an example. But for your docking and other things, did you select something from the plant yourself? Or?
Sir, no, sir. I just take the flavonoid and terpenoid family classes and dock the compounds.
Okay, so you just did a docking of the already available structures of those.
Yes, sir. Yes, sir.
And so what is your major sort of conclusion or finding or learning from this exercise?
Yeah, major finding is that sir, flavonoid class have a more activity as per the selected ligands, and we want to further future studies on the other flavonoid family. Since flavonoids already have some cardioprotective effect, we are expecting to assess the activity of the plant.
So your ligand is the BK channel proteins.
Yes, sir, BK channel.
So what are these BK channels and how are they important?
BK channels are big conductance, calcium activated, potassium channel. Actually, it's potassium channels, sir, but it's get activated by the calcium. And after the potassium efflux, they inhibit the calcium. And this can be used in the case of cardiac ischemia. By giving the BK inhibitors, we can produce BK inhibitors. We can control the influx of the calcium ion and the smooth muscle contraction. So BK inhibitors can be used in the case of cardiac ischemia.
Okay. 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 Thank you, Dr. Archana.
Thank you, sir.
Yeah, Dr. Angatha.
Archana, sir. Archana, ma'am.
All right. Just a small question. Why have you used only one software for docking?
We initially started the work only, sir, ma'am, so that we go for only one software. We will look into further, ma'am.
Okay. All right. Thank you.
Thank you, ma'am.
All right. Thank you, Archana. And
Thank you, ma'am.
thank you. With this presentation, I think we have come to the end of this oral presentation, and there will be one best presentation that will be adjudged by the that may have been adjudged by the judges by now. And I would like to express our heartfelt thanks to the judges, Professor Sandeep Das and Dr. Babita Kola, for sparing for sparing so much of time from their schedule for judging this event, and being so patient in listening to all those presentations so patiently, and asking the questions, and participating so actively. Thank you, sir. Thank you, ma'am. Now I would hand over to Dr. Ratnam Vatal for the further proceedings.
Thank you, Dr. Tabassum. It was a great session, and I'm sure everyone enjoyed it. Now, before we go on, before the results come, and I would like to request everyone to switch on their cameras so that we can have a group photograph. So everyone, switch on your cameras. All the participants, resource persons, guests. There's one Sanjay Kumar guest. His camera is not on. Quite a few cameras are not on. Yeah. Different frames, please. Done? Dr. Ruchi, Dr. Savinder?
Yes, ma'am. I have taken some.
Yes, ma'am. It's done.
All right. Thank you, everyone. Now, by the time the results are being worked out, I think if anybody wishes to say something about the conference, we can have them unmuted and they can speak. Anybody wishes to say something about the conference?
ठीक है श्योर मुझे इसी पे भेज दीजिएगा ठीक है
Abharna, what is your take?
Ma'am, first of all, it was a great experience for me joining here. I don't know how far I was successful in imparting the resource, in the hands-on session, but it was really a nice experience, ma'am. And it's a very nice lesson for me as well. Yeah, it was a very nice experience hearing all these talks. But actually, the problem for me being here is the time difference. Also, although I was trying to adjust it, but I could adjust it at the end, ma'am. And it was so nice seeing all of my teachers once again. So
good. We also felt very happy when you were on the other side of the table, not as a teacher, not as a student, but as a teacher. So that is it. Anyone else would like to, any other participant would like to say something till the results are finalized?
Ma'am, there is one announcement.
Yeah, I'll do those.
Okay.
We will be announcing the awards, of course, in due course of time, but we'll also be circulating the feedback form and maybe by tomorrow. And please do send us the feedback form, because this will help us improve ourselves and look into the shortcomings of the organization. Also, what you would like us to do in future, and what are the changes you would recommend. Second thing is that the certificates will be prepared now, and it may take about 2 weeks or little more. So please be patient about these certificates. You will definitely get them, but it will take a while, because as you know, in Delhi University, teaching, exams, invigilation, evaluation, everything is going on. So it may, retard the speed of coming up with it. So that is it.
दी मैं आपको 5 मिनट में कॉल करूं?
Any other
Yeah,
announcement?
ma'am, actually,
Yeah.
I'm sorry to say in
Yes,
between,
honey.
but I just wanted to say hello to Savinder sir. He was my teacher when I was in Hindu College, Delhi University. It was maybe his first class as a guest lecturer, and while he was still defending his PhD thesis, I still remember he given us a party when he defended his thesis. So I thank you so much, sir. It's very nice to see you once again. Thank you, sir, and thank you
Honey, your
everyone.
presentation was very nice. I really liked it.
Thank you, mom. Thank
Yeah,
you.
and it's good to see you again after so much time.
Yes, sir. Thank you, sir. Thank you, mom. Thank you,
And
everyone.
your batch is one of the most memorable batch for me because it was the first batch for me as a teacher. And I still remember most of your name.
Thank you, sir. Thank you so much, sir. I still remember, sir, you used to teach the pasta sequences. We used to have this bioinformatics class, and you used to. So it was. Thank you so much. Thank you, sir.
Anyone else?
Thank you, dear.
Professor Das, results are ready. But the competition is too tough. I felt the competition was too tough.
No, I sent my result already to the other judge.
Okay.
Yeah, I think it's with
So,
Dr. Bobby.
Professor Das, why don't you say something about the competition, the presentations, and anything that you would want us to improve upon?
So I think within the limits that we had of online network issues and people connecting from all parts of India, it was very, very well organized. Because yesterday I saw most of the teachers here in the department doing the other duty of, looking forward to NEP 2020, and yet the conference was not disturbed. It went on as scheduled. Participants were not inconvenienced. It is a great achievement. So probably it is the blessing of being online. That could be one positive side of online. But I really missed being meeting in person with the participants, interacting with the participants. Because today I realized that I was facing a lot of this disconnection problem because I'm working from the office. This has been a challenge for me, and I understand what it must be for you and all the participants. So that's the general comment. I always have some bias towards Zakir Hussain from being from there. And I still remember when we were students, I think ours was the first batch in 1987 to move to the new building. All the administration used to be in the old building where hostel was, where Professor Virendra Kumar used to take classes in his warden, when he was warden in his house. So those are memorable times for all of us. We would go on field visits. Professor Virendra Kumar would take us, just walking from Zakir Hussain College to Rajghat and other places to look at, plant. So hands-on field experience. Dr. Harjinder Grover, I think that time she was the senior most after Dr. Virendra Kumar. So a lot of interaction with Dr. Dr. Harjinder Grover, then Dr. Sunita Puri, subsequently Dr. Aisha ma'am, Dr. Vishwas, Afzal sir, Mr. Prithpal. So we have lots of, these server ma'am, when she used to be in botany library, subsequently she moved to Jamia Millia. So I don't know, I mean, Dr. Mrinal, Bhargav ma'am, Rita Khosla ma'am. So we have lots of, reasons to be biased towards Zakir Hussain. I have reasons to be biased towards Zakir Hussain. But anyway, it was a very well organized thing. And I still, because the first day when I finished, I realized that there was a hands-on training program being done by students of the RNA sequencing analysis and all those things. So I could not stay for all these sessions, or the sessions I thought I would sort of participate, at least attend some of those few lectures, but unfortunately I could not. But I hope that the college takes it up again, and we are able to do something more offline in maybe a few months time, or maybe next year, something to do. But yeah, I enjoyed it. In fact, I was looking forward to not the first, and even today. Today, some of the presentations were really good. I mean, I was very impressed by the presentations, especially, unfortunately, or especially by the PhD students. There's a lot of talent that's hidden in PhD students. The one, I think, one student from IIT, then Archana Raj, who presented at the end, and even I think one UG student who presented in the first one. So there is this enthusiasm among students to as teachers, we always tell them so many things. You do this, you do that, you do this, you do that, and we, in our own enthusiasm, we overload them with so much. But students, I think we must realize that they have an unending source of energy. And as a teacher, we must channelize that. So towards the right cause, towards the right path. And today it was on display. I mean, the presentations were very good. So I think I'll stop here. Maybe Dr. Babita has already compiled and she's looking forward to sending it. Yeah.
Thank you,
You
Professor
have to, Dr. Babita, you have to unmute yourself.
Sorry. Professor Sandeep, I have just sent you the results. Just have a look at it.
Okay.
So I think we can announce the awards now. And can I request Professor Das to announce the oral presentation award, the best oral presentation? Professor Das, can you hear me? Am I audible?
Yes, you are audible, ma'am. Probably network connection of Professor Das is a bit down.
Okay, we have, then we can announce the poster first.
Yeah. Yeah, meanwhile,
And,
he will join, I guess.
Yeah. So I hope Dr. Gaikwad is here. There were two judges, Dr. Gaikwad and Dr. Ansari, for poster presentation. So is Dr. Gaikwad here?
Yes, good afternoon, Dr. Ratnam. I'm there.
Yeah, good afternoon. Will you say something about the competition and announce the award also?
Yes, I'd be happy to do so. So I was really very impressed by all the poster presentations which were there. Though the list was long, the presenters were a few. I would have loved to hear all of them. But we judged the posters on certain criteria, which was the preparation, the presentation, the novelty, and how they comprehended the information. So, though we have given importance to the ones who have presented their own work, I totally understand that it is not possible for everybody who participated at the stage that they are to be to be able to reach a stage where they could present their own work. So, the reviews that were presented, I have taken into account, like we both decided and we took into account how well the candidate has been able to comprehend the information, because that is the first step in any kind of research. How well do you read the review? How well do you read the literature and review the information, interpret it, know what is already there and what steps you have to go ahead further? So that in itself is the first step, well begun is half done. So that is what we took into account. And though I had communicated on these basis, though I'm sorry, there was again, as Dr. Das said, some issues in my workplace also with the internet connectivity. We had the server that was down for some time, for which Anjana had to repeat her presentation. Sorry about that, but though I have communicated to Dr. Vahid, I'm sure it's the same. If you can quickly give me a quick yes to it, because we did not correspond after that. Then I'll go ahead. Is Dr. Vahid around?
No, ma'am, sir is not there. You can
Okay.
go ahead and
Yeah, I'll go ahead. I mean, he had given me his interpretation first, and then I seconded it and made some modifications.
Best two.
The best two?
Yes.
Okay. So I'll go ahead with the best two. Sanjay Kumar, Pradipta Paul, Pradeep Yadav, Riddhima Kaul, S.S. Maitra, Saurabh Kumar Jha, Ali Chari, whose work was presented as glycosylated bioactive flavonoid inhibits SARS-CoV-2 proteases and human transmembrane protease TMPRSS2. A molecular dynamics simulation study stand first, followed by the poster presented by Dr. Anjana Singh, Nikita Kritika Shukla, and Salik Nurani Khan, which is a detection of known and novel plant viruses using next generation sequencing. So this group stands second. So I congratulate both of you on presenting this information very well, very eloquently, and very well presented in the form of a poster. And all the best in addition to all of you. Rinku, who was talking about DNA helices, the potential role of DNA helices in providing salinity tolerance is also very interesting work which she pulled out and was discussing. That again was nice, and so was the microRNAs. The role of proteomics in crop stress tolerance. So all the work, I mean, all the presenters did a great job. All the best to all of you. But yes, somebody has to be the first and second. So this was it. All the best to all of you. Thank you.
Thank you, Dr. Gaikwad. I think the competition was tough and less number of presentations because, as I said earlier, exam time, evaluation time, teaching time, conduct of exams, everything is happening simultaneously. So participation they did. Huh? Sorry?
NEP. NEP.
NEP time, right? So that is why a lot of them gave, but last minute sort of couldn't make it. So thank you once again.
Thank you. I would also take this opportunity to congratulate all the organizers and beautifully coordinated event, very well managed. And yes, thank you for making me a part of it.
Welcome. Look forward to another opportunity to take you in. I'll go to Professor Das now to announce the awards for oral presentation.
So first of all, thank you for this opportunity. It was a time that was very well spent listening to exciting research being done all over. I think with Dr. Babita and me, so we had some, before we announced the results, we had some basic criteria. One was the presentation skill of the student or the presenter, which included, the slide organization, the clarity of thought, the way the slides were explained, and whether the presenter was able to stick to the time that was allotted, because these are all part of communications that is meant to be in a presentation. So that was one part, and the second, of course, the clarity of thought while you're presenting, and subsequently when anybody was asking any question, whether there was a hypothesis to the research that was being done, and so on and so forth. So based on those two, and as has just been said, that unfortunately life is such that some people will go or stand on the podium, some will stand on the silver medal, some will stand on the bronze medal, but everybody is actually a winner here. But together with Dr. Babita, we have actually two joint first prize winners. So I will announce one and Dr. Babita will announce the second one. So one goes to Ms. Archana Raj and Suresh Nair on their work on in silico molecular docking and pharmacokinetics of selected phytochemical against PK channel. So that's one of the joint first winners. Dr. Babita will announce the second one.
Yeah, the second, do you have any guesses? All right, I will not hold back this the result. It is Bani Konkam and Hari Prasad Puttaswamy, who presented their work on computational studies to evaluate the SHV1 beta lactamase in Ficus religiosa. And I would say, as Professor Sandeep has already said, it was a delight listening to each one of you. And yes, there has to be, somebody has to be first and somebody has to be second, and that's how it is. But we would say that we have really, really thoroughly enjoyed the presentations, and what a concept clarity and the new ideas that you have brought forward. And that is an eye opener. And many of the works that you have presented, they are much required at present times. And I'm sure you're going to take it further. And I would like to congratulate all the participants, be it oral presentation or poster presentation. And best of luck for the next time. And I would also like to thank organizers for giving this opportunity. And it was a delight to, work with my own lab mate, Professor Sandeep. So it reminded me of old times when we were together. So giving this opportunity was going back to those times. So he has always been a humble, and he reminds me of Professor Shivanda. I feel somewhere that image of Sir is there in him. So it was a delight working with you. Thank you so much.
Thank you all. Thanks to you.
Thank you, judges, for sparing your time. Now it's time for formal vote of thanks. And we had, the 3 days event, I think, went with a just flying time. We had an overview of the theme of the conference, covering essentially use of omics approach in wide variety of fields, ranging from crop improvement, where Professor Sandeep Das explained the use of the technique in the members of Brassicacea. We had another very beautiful lecture by Professor Ute, where she explained use of omics approach to combat environmental challenges. And there was also talk on protein corona, a tool for understanding nanoparticle biointeraction in the field of phyto-nanotechnology, a very impressive talk by Professor Renu Deswal. Studies also explained use of technology in the study of, the lectures also explained the use of technology in the study of plant proteomes. And the last two lectures were highlighting the in silico approach for drug designing and drug discovery. And beyond that, we had the wonderful hands-on sessions given by some of our very bright young researchers. On the whole, I think the event had a lot of information to take home and tremendous food for thought for those of you who wish to enter the field of research. I feel this conference introduced our young students to change of mindset in terms of research by, from wet labs where we actually take test tubes and work with it. And on the other hand, in silico research, which does not require actual experiments, but use the data by the wet lab to analyze and use the technique of omics. I think with this overview, I would now go to the formal vote of thanks. And I, on behalf of the organizing committee of International Conference Cum Workshop and Department of Botany, Zakir Hussain Delhi College, would like to thank our esteemed speakers, Professor Lakhanpal, Professor Das, Professor Ute, Professor Renu Deswal, Dr. Ravi, Dr. Vinay, Dr. Brijesh Rathi, for enlightening us with their knowledge and experience on the theme of the conference. We are grateful to them for having accepted our invitation and spared their valuable time. In fact, some of them had to really, push. You saw Professor Suman Lakhanpal, squeezing time out of the meeting. And then similarly, Professor Ute also was a little short of time, but somehow she managed to give a wonderful talk. Thanks are also due to the very lucid hands-on sessions by our young researchers. These sessions were indeed the very crucial segment of this 3 days conference. We are indebted to our judges, Professor Sandeep Das, Dr. Babita C. Kola, Dr. Wahid Ansari, and Dr. Ambika Baldev Gaikwad for their time and efforts to judge the competitions. We also wish to express our gratitude to the participants for their interest in the conference and participating in the oral and poster presentation. The organizing committee wishes to put on record the unstinted support achieved from the head of the institution, Principal Professor Narendra Singh, and head of the department, Professor Suman Lakhanpal, in the conduct of the 3-day event. The organizing committee wishes to express their deep sense of gratitude to all the members of the Department of Botany, Zakir Hussain Delhi College, for their active participation in smooth conduct of the event. And a very big thank you to all our undergraduate students who sustained through 3 days of this conference, knowing very well that probably it was going much above their head, but I'm sure the subconscious mind would have absorbed some of the points, some of the words, which will come handy later on. Thank you all once again for being with us this afternoon. And once again, I remind you, certificates will be sent to you in due course of time. Please do not forget to fill up the feedback form. And with this, we'll close the event. And please stand up if you can for the national anthem that will be played online. Thank you.
Vindhya
Thank you so much. You may now log out and have a good day, everyone.
Thank you, everybody.
Thank you all. Bye-bye.
Thank you, everyone.